Garrett,
Yeah,
good to good to catch up with you here.
yeah, nice to see you, Cory.
I I appreciated that note you sent me recently too, because I've kind of just done a few of these recordings and stuff just to try to capture some educational content,
Yeah.
and pick apart issues or pick apart ideas and just talk at them in length. And
hmm
it was kind of cool that evidently some of it had some use in
Yeah.
your eyes.
Yeah. I mean, I like the whole informal speaking format that you to have here because it's literally most of the time it's like two beekeepers
Hmm.
or two people sometimes, but a lot of them are beekeepers sitting down and just having a chat, and it goes wherever the conversation leads. And there's nobody like on your the one where you're talking about inseminations, and you're discussing bad with names, so I can't remember his name right off. Discussing, oh, I've had this that problem, and what do you do for this? And you know, I train people on this occasionally, and it's nice to be like, hey, look, so you're not the only one that this is common with learning this technique.
Yeah, there's so many intricacies to it, even because I think it was 2014 maybe, whenever I learned from Sue. And it's not like I do it all year long or anything, but I've got several years of experience and still head scratching stuff happens, like I don't know exactly why, or did contamination occur somewhere? It
Yeah,
just seems like it's always something, and I'd just like to have a realistic outlook on things.
yeah.
Uh,
Well, I'll tell you that even after 30 years, you still have those moments.
yeah,
So, I mean, there's just things that happen and you can't explain it and
yep.
you have no idea.
And Sue had spoke a little bit about that too. I had a conversation with Sue Kobe about instrumental insemination, and same thing, she was trying to keep some warm and accidentally whacked some queens, and but and she's the master at it, she's
Yeah.
internationally
But
known. But
yeah,
I
she
I
trained
just
me.
appreciate.
So,
Yeah, that's what I'd heard. Did you
yeah.
learn that was it when she was still at Rothenbuehler lab? And
Yeah.
okay.
Yeah. I was a student worker there. Well, actually,
Okay.
what happened was I was going for entomology, and I took their beekeeping biology course, and
Okay.
or the bee biology course. And so part of that is, hey, you go to the lab and you actually go out into bees, and then you go. It's on the outskirts of camp on the farm complex, and so there's fields and all this stuff. And we had to take do a Hamanatra collection as part of the project. Well, then I would just wander off and follow Sue around, because Sue would go and say, okay, here's the beehive, blah 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 blah, and I would go follow her around. And then once she realized that I knew somewhat of what I was doing to a small degree, I'd been keeping bees since I was 14,
Oh,
so
cool.
I knew beekeeping, to
Mhm.
a certain extent. I mean, when you get somebody, that's been doing it for years and decades, then you your little bit kind of seems little when you think about it.
Sure.
But I started basically working for her as a student worker during the summertime, and she offered, she offered her insemination class, and I said, hey, I'd be interested, and she was like, well, that's it's full, but you can sit and listen, and we can work on it on the side when there's gaps, and then if I need you to go get drones or to do this or that, you need to do that. And that's when I learned. So I learned back in 93.
Did you
I think it
really?
was. Yeah.
That's awesome.
Yeah. I think it was 93 or 94. I think it was 93 was when
So
I learned.
you mentioned entomology. You were studying at OSU then for entomology?
Yeah. For a short extent.
I was just going to touch a little bit kind of on your background, like where you came from and, education. Obviously, you've got hands-on beekeeping experience.
Yeah. Well, I'm from Ohio originally. My whole family's there. Uh, like I said, I caught my first swarm when I was 14. My grandpa, yeah, I was interested in in bugs. And so I was like, oh, I'm going to go to college for entomology. This is great. You know, but in junior high, I was like that. So this was the spring of my freshman year. There was a swarm landed in my grandpa's apple tree. And he was like, hey, come look at this. Come look at this. So I go, well, that's cool. And he goes, you want me to show you how to hive it? And I was like, yeah, sure. And my uncle used to have bees a while back and got out of it because of, you know, bad back, which
I understand that.
happened. Yeah, so there's some old equipment back on the farm. And so we went back there and grabbed a box, and literally we took a inner cover because we didn't know what we had and what we were doing. So we took an inner cover, nailed a small piece of wood over the hole of the inner cover, notched out a hole on the rim and nailed it to the bottom.
Mm-hmm.
Then found the lid and took it. And I think we had some old frames, but you know, no wax or anything. This was before plastic, so it was just the frames with the wire for all on how they want to draw it. And you know, we took it out there and shook the bees in, and that was that. And that started the long journey of, you know, once you do that, you're hooked, and it's just bees, bees, bees. I went to Ohio State for entomology because that was, you know, the only place you could really go in Ohio for an entomology degree. You know, they're rare universities to find where you can get that. So I went there. I also in the summertime would do county bee inspections for the apiary service.
Really? That's cool.
So I started that in high school because, you know, it's just a summer thing. So it makes a nice summer job. And so I would do that. And after a while, you know, I met my wife, got married. You know, the degree was coming along, but I had to pay for it myself. So that meant I had to work, pay for it off to the side. And eventually, you know, life happened, and it was like, okay, I'm just gonna work.
Yep.
And then, you know, had kids. Later on, it was like, you know, I want to say it would have been about 2005 or something like that. It was like, yeah, I should go back to school. You know, I'm getting tired of this. I had a retail job. I was a manager at the grocery store in the meat department. And so, you know, that's weekends, every weekend, every holiday.
Oh yeah.
And, you know, my wife was my token, you know, person to my family, you know. You know,
Yeah.
they never saw me, they saw her. And then I heard what was going on with the family through her. And so it was like, maybe I should go back. And since I was already into the business field, I actually went back in night classes for business. So I got my business degree.
Cool.
And right after I got my business degree, I was, you know, trying to decide, because I'd quit my job eventually and tried a different job, you know. So I was like, okay. Now I was a stay-at-home dad for, you know, 6 months or something like that, which was great with, you know, the little kids, you know. But you know, eventually I was finishing up my degree, and Sue called me up, says, hey, I hear you're going to school and not working a whole lot. Can you come and help me? I've got a lot of trips this spring, and I need help with the beekeeping. You know, she had a couple students, but you know, the whole, you know, beekeeping is not something you can learn and here, let me go show you and go do it type thing. Yeah. So I went back and helped her for the spring, and then on through the summer. And then when winter came, she was like, okay, that's, you know, that's good. I don't need you for the winter. And then the next spring she comes up and says, hey, I'm moving to Davis. Can you come back and help me pack everything up?
Yeah.
So I was like, yeah, I can do that. So, and my packing everything up was more like going out and caging queens to be shipped to Davis for their breeders. And then the entomology department there asked me to stay on to take care of the bees until they figured out what they were going to do, because you know, the beekeeping or the bee scientist, he left for Arizona, Brian Smith. He left for Arizona a couple years earlier than Sue left. So they had nobody to take care of the bees.
Yeah.
And then they have the Worcester lab that had Jim too there. And, they were just trying to figure out, we need to, how are we going to work this? And so I stayed on, and then Sue contacted me and said, USDA lab in Baton Rouge has an opening, and I think you should apply. And I was like, okay, that's good. I don't know whether I really want to move to Baton Rouge with my family and all that stuff. So that's, we're all right from that area. And I was like, but I'll apply and see how it goes. And I got an interview, got the job.
Awesome.
And I was like, okay, I guess I'm moving to Baton Rouge. And so, it'll be 15 years ago this fall, I moved to Baton Rouge and started working at the Bee Lab, working with Bob Danka on the VSH bees.
That's awesome.
So, the funny thing about that was my first day, I went there and it's like, okay, here you go, set up your instrument because we need you to do some inseminations.
Really?
Okay. Where am I going to set up at? Well, you can just set up here at this spot here and use this microscope. That's where John Harbaugh always did his inseminations. So
that's awesome.
Oh, I'm sitting in John Harbaugh's spot.
Big shoes to fill.
Yeah. And what Bob did was he said, so I've got a queen on the other side that is a virgin. I gassed her twice to get her to lay drones.
Mm-hmm.
And I want you, and I caged her. So now I need you to go grab her and inseminate her with her own drones. That's what I want you to do. So that's what I did. It got really weird after that, because then we raised daughters off of her. Then we dissected her spermatheca, collected the semen, and inseminated her daughter with the semen.
Oh, wow. That's crazy.
I don't know what the purpose of it was now, but it was just something he was trying to inbreed something for whatever reason. And but it was just one of those, like, the weird stuff you can do with insemination.
I haven't done that yet.
Well, it didn't work very well. There's not a lot of semen in the spermatheca, especially because it was a single drone insemination.
Mm-hmm.
If it was a multiple, I think you can get enough semen to actually and take this semen out of the spermatheca and inseminate another queen, enough to get workers, so that maybe you can actually, eggs that you can actually graft from.
Sure.
Or something like that. But, it didn't really work. I think I got, most of it was drone layer. She was drone layer, but it was like, oh man, I'm going to be in for some weird stuff going on here.
Right.
For sure.
That's your first day. Yeah. I'd be thinking the same thing. Man, what do you think, like, as far as VSH goes? I know, like, it's not new news, but I feel like there's a bit of a revival maybe going on, or a renewed interest in it because of, like, amatraz resistant cases, or at least popping up, or suspect anyway. It seems like, which, from entomology, host resistance is the cat's pajamas. That's where it's at. And it seems like there's kind of a renewed interest in that. But what are your thoughts on it from your side? Same old, or?
Yeah, well, I mean, there's a lot more going on. Of course, Harbaugh started, discovered what at that time was called SMR, for suppressive mite reproduction. And we found out that it was hygiene, so we changed the name to Varroa sensitive hygiene. So this has been going on since the late 1990s, I think.
Yeah, I was thinking 95 maybe is whenever he figured it, or, well, I don't want to get my wires crossed. Was 95 when they asked the Bee Lab to try to breed for resistance, maybe? Or was that?
It might have been, because it was kind of like the Russian program and the VSH was kind of going on, started around the same time. Because the lab was basically told, see if you can breed resistance to Varroa.
Yep.
And so, well, we're gonna, the one thought was, well, we should go to Russia and grab the bees that have been associated with Varroa the most.
Mm-hmm.
And another one was, well, let's see if we can breed any resistance with the bees that are here.
Yeah.
So, at that time, they didn't even think resistance was possible.
Yeah.
I mean, yet you heard all these, at that time, it was like, you can't breed for resistance. It's like breeding sheep to be resistant to wolves.
I've heard that. Yeah.
And so it was like, is it going to work? Nobody knows. So, the lab was smart in that, as they took two different viewpoints to see if it would work. And it just so happens that both of them worked.
Yeah.
I mean, the Russian bees have resistance to Varroa. And then on the other side, we found VSH, which we found a particular trait that controls Varroa.
Yeah.
So, that was really interesting. I came along, after Harbo was gone, and the VSH line at the lab was pretty well established.
Yeah.
Tom Glenn had an agreement to sell VSH inseminated queens to the bee industry. We provided him with the stock. And of course, he provided us stuff back just, so we could test it and, incorporate it back in, trying to avoid inbreeding to a degree. But the stock was never, at that time, it was never a line of bees. It was bees just holding a trait.
Yep.
Oh, yep. Okay. You're breeding other bees into them and you're selecting for the trade. And, there was no type of bee, a VSH was a trait. And that was the way John Harbo envisioned it, was we sell these breeder queens that have very high, because at that time it was, what we had was very high. I mean, you heard stories of they cannibalized all the brood.
Too hygienic or whatever.
Yeah, too hygienic. They're extreme OCD, all types of stuff like that. But Harbo's idea was, you sell those to beekeepers or to bee breeders. They raise daughters off of them and made them to their stock, and they have resistance to, an average good degree. And he did research to prove that, you can made it to non-resistant stock, and they have good enough resistance that you don't have to treat. At that time, I think everybody was treating at least 2 or 3 times a year, up north, let alone down, what they did down here in the south. But how it happens, they get these queens, and they're pulling the brood out all the time. The colonies didn't grow. They're like, the patterns look like crap because they're pulling all the brood out, and they're like, I'm not grafting off of this. This colony is terrible.
Yeah.
So that didn't work. It just we couldn't get it through that this is just the trait, not a line of bees. And so we had to rethink, and that's where we came up with what's now called the Paul line, which is like the new VSH at the lab. And that is more of a stock of bees. So what we ended up doing there is we ended up, asking commercial beekeepers, all the big guys and all that stuff, what are you looking for in a bee? Mite resistance, disease resistance, good honey production, strong buildup for almonds, and at that time, a yellow bee.
Yeah.
Because all those traits are Italian traits, and so therefore the bee has to be yellow. And so it was like, okay. So what we did was we took VSH virgins and made them into 3 different cooperators operations.
Yep.
They just let them go out there and mate, and then we had their. So it was kind of a study to a degree, but also a breeding practice where we had their strand of bees. So we had colonies that half of the colonies were their bees and half the colonies were VSH mated to their bees in a sense. And we would go in and, in the fall is usually when this happened. Well, I guess no, it happened in the spring, and we would mark and all the queens, verify that they're there, take a mite sample, population count, general stuff like that. And then we said, okay, they're yours. And then we waited until they came back the next spring from the almonds, and we went through, verified if the queen was there, and if took a population count, and then took another mite sample, and then we chose the best ones.
Yeah.
And they had to be left untreated.
Okay. Awesome.
That was the key. They had to be left untreated, and we were doing numbers in the realms of 300 colonies.
Nice.
Per operator.
Yeah.
So, for an operator to say, I'm going to basically potentially let 300 colonies die.
Yeah.
I mean, that was,
it's a big deal.
Big deal. I mean,
yeah.
I mean, some of them wasn't as big of a deal. You got 20,000, 30,000 colonies. That's not so bad, but you got 3,000.
Yeah.
That's a big deal.
Yeah.
So, and of course we found good stock. We brought it in. And then I would do inseminations with it. So we had all these queens, and I would inseminate them, kind of forming a closed population. But then what we would do for the continuing generations in these cooperators is we would breed, take pure VSH virgins again. And we would take these ones that we pulled out from the previous generation and raise daughters off of them and bring them out. So you basically had something that was a version that was 50% VSH, 100% VSH, and then their stock, which was 0.
Yeah.
And the whole point of this was kind of show that if you do this, what Harbo was suggesting was right. You just need to take some breeders and buy them for several years in a row and keep reintroducing them.
Yep.
And take your best, and you will be able to have your type of bee with VSH.
I love that. And he's right, because it's an additive trait, which is, he said it was an additive trait. It's like you said, some people I think VSH is like an Italian or a Caucasian. It's just strictly a trait set. But that's kind of what I've done too, which I've used Harbo's VSH assays. And that's my point too, is like, don't just rely on me necessarily for it. Once you get it and you stick it in your population, then I put a video out it was just Harbo's VSH assay, to where you can keep that selection pressure on it and come up with a wherever you're at that does well or does well with your management style. And you can fix that trait and keep pressure on it too, to where you can keep it high enough that it actually does something. And I know you mentioned Tom Glenn too. That was my first experience with VSH. And I know some people kind of have a bad taste in their mouth, like, oh, I tried them and they weren't any good. But who knows where they came from, or if they were an F3 or F4 or F5, or if they even had VSH. But Tom Glenn's, his were awesome. Like, I just had XYZ random bees, I would see deformed wing virus, I would see chalk brood, I would have crappy winter survival, and that was with trying to manage mites too. And then I bought his breeders and started raising queens from them. And anytime I saw any issue, period, I would requeen with one of Tom's VSH Italians. And my whole operation did a 180-degree turn on how it looked and how it was productive too. And so I had a different experience. I had VSH I thought were extremely good and were productive bees, and I've been kind of stuck on it ever since.
That's good.
Yeah,
I think because I mean, it really is. The Pauline population. I don't treat any colonies at the lab unless they are queens that we purchase from a queen producer.
Sure.
We keep a variety of stocks around to use as controls for experiments, or if we're breeding, like we're messing around with looking at breeding for resistance to like deformed wing virus.
Yeah.
Or stuff like that. And which could be very rare, and so you need a variety. You just can't say, well, we're just going to take Russians and do this, because they might not have all the traits. The same with Pauline might not. So and you want to hedge your bet. So it's like they look at everything. Basically, what I do is I do tons of single drone inseminations. I'll do 80, 90 and single drone inseminations of, and it's all mixed up. It's this one crossed with it, just whatever. And then they could try to see if they can sort that out and carry it through a couple generations. And, sometimes it works, sometimes it doesn't. Sometimes it's working. And then you have this whole, how do I overwinter single drones because it's not far enough along. And so you try and, you lose most of them.
Yeah.
So
how long do they last typically?
It really depends on how good the insemination is.
Okay.
But I typically say, plan on about 6 months.
That's not bad. I mean, that can give you some time to do some measurements and figure out what they've got and what they don't.
Yeah, it gives you time to let the colony switch over completely to her bees and be able to do some type of a evaluation or measurement of whatever you're looking for. They can last longer. I mean, I've had them last over a year.
Dang.
That's
awesome.
That's rare. I mean, that's
that's an exception.
Yeah, but we've got some right now at the lab for an experiment that was going on that I inseminated, I think last June, and some of those queens are still around.
Still kicking. Nice.
Some of them are, 10%, 10% of their eggs that they're laying end up being drones, so they're becoming a partial drone layer. But there's still something going on, and some of them still look good. I mean, it's just one of those things. Like, I wish I could figure out how I did that insemination to get all that semen in there that she's still going
every time,
so I can duplicate it every time. So then, if you can keep these things lasting a year, that makes it a whole lot easier for these, because basically what we try to do with this is do them in the spring, get the evaluations done fast enough that you can do another generation in the fall, so that you can overwinter them and jump on them in the spring to hopefully be able, before you lose too many, to be able to do your evaluations to do the next generation.
Right.
And that takes a lot of work.
Big time. Absolutely. And like you said, you're going to have a rate of attrition too. Not all of them are going to make it. And it's just the way it is.
Yeah. I mean, they're single drones. I mean, some of them they try to supersede continuously.
Yeah.
Others they don't. I mean, it's a mixed bag. But for research purposes, for finding a particular trait or locking down a particular trait.
Oh yeah.
It's unbeatable in what you can do with it. So
yeah, especially if you're going into just an unknown population and you're looking for something that's minute or is in maybe a small percentage of it. I mean, you can get to it quick with a single drone insemination because you don't have all those other subfamilies you got to sort through.
Yep.
I've never done it. I wasn't familiar with the concept until I read Steve Tabor's Breeding Super Bees, I think, if that's the book. And he called them just SDIs, but that blew my mind because it's incredibly intelligent as far as narrowing, traits down, which I got the trait fixed. And then I kind of went into more like a Page Laidlaw closed population breeding model, maybe a variant of that, because I just knew the basic concepts of it. But I basically just raise a ton of F1s and as many breeders as I can possibly winter. And then I'll test all the breeders and then my top F1s. And then I'll graft out of the breeders. And then I catch drones from, maybe an entire yard, 50 colonies upwards that score high enough on Harbo's assays that I want to keep them and I like them. And then I just keep rotating and keep the process going. But I mean, I've got a day job. I wish I just could play with bees, but I haven't been able to make it work financially yet. I love it, but anyway, I keep less than 200 colonies. I've got enough wood to have around 200, so it's not a bad sample size, but like you were saying earlier, if you could work on a larger scale co-op with some people or something to where you've got just a big wider net.
I mean, I mean, the larger you can do it with smaller, because I mean Page Laidlaw post population basically said, anywhere from, depending on how you do it, 15 to 20, 25 breeders per generation.
Yep.
And it's going to last. And that's if you just use those queens and their drones.
And never outcross anything.
And never outcross anything.
Yep.
You look, it should last for at least 20 generations.
Yep.
So, the more, and I look at it like that too, the more drones sources I can use, I think that, I can limit the queen source a little bit.
Yes.
So I have a larger drone source. So I do that. The poll line at the lab is done a very similar fashion. I select the best, around 30 queens that I have, and they're all inseminated. I mean, by the time I go into winter, I maintain around 300 colonies myself at the lab. The lab itself maintains around 700 or 800.
What? Are you serious?
Yeah.
Oh my gosh, that's amazing.
And then in the summertime, it can be a lot more, because there could be a lot more nukes around with SDIs or just colony split. We lose a bunch. I mean, we need Varroa for experiments, so you bring in commercial stock and you let the Varroa die, Varroa, and then the colony dies.
Yeah,
it's like, I just one more time and I'll treat it, and then by the time you get ready to treat it, it's too late and they're dead. But that's what I do with the poll line, is I pick out like the best 30-ish is what I do, and then I narrow that down to the top. Usually I try to hit around 15.
Yeah. Awesome.
So I have 15 that I graft from, but I use drones from all 30 of them.
All of them. Yep.
And that provides many different things. It provides a lot of drones, so you're never short on drones, because that makes it really tough. And I try to do these things, because of research, matings and stuff that I have to do. I try to do all the maintenance of the stocks, as quickly as possible.
Get it out of the way. So
get it out of the way, so they're going, and then all I have to do is maintain them. I don't have to worry about them, and then I can sit there and concentrate on these SDIs, or one time I inseminated queens that went into space.
What? Are you serious? That's freaking cool.
It was, it wasn't like they stayed on the space station or anything, but they basically went up in a rocket and came down.
Yeah.
Thing. And I mean, it was something with MIT. I have no idea what the main purpose was. I was just asked, hey, can you inseminate, X number of queens? I think it was like 8 or something like that. And get them laying, and then we shipped them to them, and they kept some on the ground and looked at how they did, and they had little special cages for the queens and some bees, and
that's wild.
It was kind of cool. I think if it's not out, I think there is a paper coming out on it.
That's pretty neat.
But it's all types of weird stuff like that. But the main stuff that I'm responsible for is I'm responsible for maintaining the poll line.
The poll line, yeah.
I mean, that's most of my effort is put into that, because it's so important, with the research at the lab, and then maintaining it for, and others' research. We got agreements with different universities and different labs that we send them queens every year, and for them to do research.
That's cool.
So there's a lot of that. And then we also have the Hilo population out in Hawaii that we are working on, and that was our attempt to that is our attempt to get the VSH into a population that is commercially viable.
Yep.
At a commercial level.
Yes.
The other stuff like what Tom Glenn did, and Adam Finkelstein with VP Queens, and what you're doing and stuff, that's great. But honestly, who VP Queens then sells breeders to make a small number of queens, or they just raise queens for themselves.
Right.
Commercial
Not
guy
thousands.
not thousands to
Yeah.
sell or tens of thousands to sell. And so Hawaii is a great place to raise queens.
Oh yeah.
So we started, we kind of talked about stuff there and started that program. And that's our attempt to have breeding, because with everything that we do at the research, I mean, you saw that with the Russian bees, we bred them, and then the Russian Bee Breeders Association started up and they took over control of that.
Mm-hmm.
So we don't have to spend our resources trying to maintain
Constantly.
that constant, population. We want to do the same thing with VSH. We want to get it to somebody that can maintain it to a high level that it needs to be and distribute it out to whoever wants it, basically. Yep. So we can go and say, okay, Garrett doesn't have to spend all of his time maintaining this population. Now he can work on this and this and this that we have for him to do. So that's the whole point of the program. But involves, that involves a lot of work because, VSH trait is not easy to select for.
Mm-hmm.
You can, but it's very labor intensive.
Indeed.
And when, to go through all the brood to find it. So
I usually have people over and tell them it'll be real fun and we're going to have a party and hang out. Which we actually normally do have fun. I mean, there's some level of work in it, I think we're testing April 9th and 10th this year. And I'll have, I think, my regular group of friends that come down, but Eugene Makovic, that is the editor of American Bee Journal, he's going to come down. I've known him for a long time after serving with him on the state board, and he was interested in checking it out. So he's going to come down too and help us test.
Yeah,
it is definitely labor intensive.
Yeah. Well, and Harbo's assay that he developed is quite simple compared to what we do at the lab for research purposes.
Okay. How would you compare it? Like the one that you guys do, what's the main difference? Just a lot more bigger sample size, or?
Yes. So what we end up, our normal standard protocol, now, granted, we really haven't done much of this since the pandemic. The lab has been closed down since, whatever, March of 2020.
Yeah. I think I've been remote since 2 years this March. So I bet that's probably around when, March 20.
Yeah. So, it's basically the work that we're done there is maintaining, basically it's just maintaining bees and what they call essential research, which has basically been, we got a grant for this and we have to use the money up by here. So we have to do this project.
Have to do these things. Yeah.
Yeah. And so, some of the other stuff has been put on hold, but typically what we end up doing for VSH is the standard has always been, we go through, you go through at least 200 cells. If you find 15 mites, you can stop. If you don't find 15 mites, you go up to 700 cells.
Oh, wow.
If you find, or till you get to 15 mites. If you find 15 mites, you can stop. I think it's 15 or 20, somewhere around there. But, I think it's 15 mites. Once you find 15 mites, you can stop. So you keep going, keep going, keep going. Once you hit 700, you can stop no matter what. On some research projects, on others, you have to go up to 1,400 cells.
Oh my gosh! Wow.
And when you're doing like the high VSH or even the pollen that we have, you don't find 15 cells. With mites. I mean, that's, if there's 15 cells of mites, that's probably one that's never going to be used as a breeder. So,
right.
That's kind of, so you end up doing 700 cells. But we did, since Bob Danka retired, I got a new researcher, which is Arion Avalos, which does like bee. My internet connection's unstable.
Yep, I can hear you again. Yeah, you kind of cut out for just a little bit. There you go.
Okay. I don't know what that was.
I don't know. It just said unstable internet connection for a minute.
Yeah.
So
Okay.
you were at, let's see, you mentioned an individual and you were talking about the assay.
Yeah. So so that was our assay, previous. I mean, that's what it was. You sat down and you may get 2 or 3 colonies done in a day.
Yeah.
Or you might not get through that many cells. You might not find brood of the right age because it was always, purple-eyed or older, preferably with tan joints or tan body.
They're a lot easier to pull out then too once they start getting tan.
And you can tell if they're non-reproductive or not a lot easier.
For sure.
You got some definite adult males, definite pseudonymphs. So, but we hired a scientist before Bob retired, but he's my new supervisor, Arion Avalos, and he's like a genomics, genetics,
Nice.
specialist. He looks at the whole genome and like does all this, magic,
Exactly.
and it's like, okay, that's great. I don't know what you did, but but he took all of the data that we had from the past for all the VSH colonies that we've tested, where we've had controls, everything. And he ran a statistical analysis on it and discovered that we pretty much can make the same decision looking at just 300 cells.
Okay. Was that why John went with, or was he just trying to simplify? Because I think he says 100, but if you don't find what you need to go to 200.
200.
To like double it. Which none of my stuff was treated easier. I think it makes it easier to see. Well, obviously you could get a false positive, if you just hit them with amitraz and there's not many mites in there, according to John's, which it'd probably be harder to fool if you did way bigger quantity like that. And it seems like too, which I'm not treating period, because I just don't have enough colonies to have a, well, this is the ones I'm breeding from. And so I kind of had to make a decision one way or the other. And it made me feel better because Jeff Harris or somebody had mentioned previously, and it may have been in a write-up over the Hilo project, but he was saying the USDA colonies hadn't been treated either, which kind of made me feel better because I caught some crap for not treating my bees. Which I was breeding specifically. It wasn't like I was just trying to manage them for honey production or whatever. I wanted to see if they actually had resistance or what happened, if I didn't do that. And so that's the route I went. But my reasoning behind it was just putting natural selection back as a actual pressure that's pushing on your bees. Was it my rationale behind it? But it made me feel good that smart people have actually done the same thing. So maybe I wasn't a complete fool.
No, I mean, at the lab here in Baton Rouge, the only colonies we treat are the commercial stock.
Yeah.
Or if it needs to be for an experiment.
Sure. Yep.
There's some experiments where it's like, we don't want any mites in there at all. So
clean them up.
Clean them up real good. Yep. But, for all the other stuff, for like the stock that we maintain, still maintain a couple lines of Russian bees, so that we're still part of the association.
Yeah.
And then we have the Pauline bees, and those never get treated.
Yeah. Awesome.
It's not even this one's got high, because that still happens. I mean, let's admit, biology, this is bees. which they're prone to wanting to outcross. They've got high recombination. This resistance is, VSH resistance is highly complex.
Very.
And so you do run across, the occasional colony that scores high. Now, how high is high? 5%, 10%, after a year of no treatment is what I consider high, but I still don't treat them. I mean, they typically don't end up dying from varroa.
Yeah.
They just get weaker, and then they end up just getting, split and recleaned. I mean, for the most part. But we don't treat any of those two populations at all.
That's awesome.
With anything, no natural oils, no oxalic acid.
That's where I'm at too. No antibiotics, no
antibiotics,
nothing.
Yep.
And the thing is too, is I've had some people that kind of are skeptical of me, which they should be. I mean, if somebody's making claims about something, you should, have a reasonable level of skepticism. But I think some people think I was possibly just full of it or don't believe, that it's possible. But even Harbo on his website said, his stuff, which it'll test super high, he came out and says it can be run without miticides. So it's not snake oil, it's measured science, but I don't know. There's still a lot of mysticism that's out about VSH and hygiene. And you mentioned earlier, where there's some that were psycho-hygienic or whatever. Inevitably, that's stuck in people's minds, even if they don't know a lot about VSH. That's one of their major turnoffs. And even though I'm showing brood frames that look phenomenal, in the springtime, it's still a concern that always seems to come up. But
I mean, we had I think it was a 3-year experiment. I think it was 2 years. Where it was a longitudinal study where we had bees that from a commercial beekeeper that overwintered in California almonds, came down south, made splits, then moved up to the Dakotas for honey production, and then moved back. And we had basically their bees and Pauline bees that we were testing, and we would go to each, so we would test them here, and we would look at what we were looking at, which involved looking at pulling some pollen, nectar, because we were looking at insecticide contamination, just, it was huge, a lot of different stuff. So we were taking a lot of different samples, but we were also measuring, frames of brood, frames of bees, verifying the queen. They measured the honey when they took the honey off for honey production. And we would do that when they were here, when they were in the Dakotas, and when they were in California. So we would, have a crew that would go to each one of these. And I think there was 300 or 400 colonies in this experiment.
Oh, cool. Yeah.
And we took mite samples. We took mite samples, and we took virus samples also. So it's we took everything. And in the end, I don't think this has been published yet, but in the end, what we found with the Pauline is that they basically maintain the same population size and honey production as the beekeeper stock.
Nice.
Which the beekeeper was allowed on his stock, was allowed to treat the colonies as he normally would.
And the Paulines no.
And Paulines no.
Awesome.
So, it ended up being, this, look, no treatment, and you're getting the same results. I mean,
I love that. That's
different
awesome.
Different years showed a little bit different things, but overall it was, for the most part, you could say they were about the same.
Yeah. I think we're getting closer, because you always hear a lot of people, oh, it'll never happen. Or like you said, breeding sheep to be resistant to wolves. That's not the case. And maybe we're not there exactly yet, which that's a lot to get the whole industry to adopt something like that, and to start maybe doing some of their own selection and stuff. But that right there is not snake oil. That's scientifically proven results that it is possible, and that we're starting to get there. I feel like,
Yeah. I mean, then it's taken a while.
Oh yeah,
Um,
absolutely. Rome wasn't built in
um,
a day, as they say.
yeah, and we've had the same problem out in Hilo with those bees, because we kept the beekeeper from, right, because he wasn't a real queen producer at that time.
Mhm.
He was on the big island is where all the queen producers are at, and he's on the island, but he's on the Hilo side, which is the wet side, and all the
Okay.
queen producers are on the other side, which is the dry, sunny, tropical like thing. I
Yeah.
think the Hilo side, I think they get like 120 inches of rain in a year.
Do they really?
Yeah. So technically for mating, it's not that great compared
Ideal.
to the other
Yeah.
side.
Mm-hmm.
But he has 7,000 colonies on that side of the island. So he pretty much has that side of the island locked down.
Mm-hmm. That's cool.
If he can make sure all of his colonies are requeened with the correct stock, then his matings will be pretty good. But we didn't want to like just start this and start selling queens, and then they're not ready yet,
Mm.
and the quality of VSH or anything else, and then beekeepers get that initial, oh look, this is crap because of this reason, and then it just keeps that
Yep.
continuing
Stigma.
on. Yeah. But we also took those queens and brought them over to the United States and did field trials for we did 3 years worth of field trials to see what they were like.
Mm-hmm.
It wasn't necessarily to show what they were like. It was for us to see what they were like and where they're good at and where they're lagging and where we need to improve on the breeding. And Hawaii, you cannot introduce queens, so VSH got there by introduction of semen. So that was so it takes a little longer to get that trait in there.
Mm-hmm.
And so initially it was done with single drone inseminations.
Okay.
And it was done, Hawaii's in a tropical region. So, we thought, oh, this is great. This is tropical. We can raise queens almost all year long.
Yeah.
There's no reason to do this whole, let's inseminate 200 queens and get them laid in this month and then watch them the rest of the time. We can do 20 this month and 20 next month, and and just kind of pace them on throughout the year. But we did single drone inseminations and looked specifically for the VSH trait, which got the VSH level there. And we were buying queens from like Kona's there, so the beekeeper would get queens from there, and we would raise, this is a really good one, so we would raise daughters off and do single drones to try to
That's cool.
bring in other genetics. So it just wasn't the pollen, we're trying to make it its unique population itself.
I like that.
And so we were doing that, and like everything, we were concentrating too much on the VSH. We weren't looking at traits like honey production. But the way we were doing it, you can't judge honey production if your population is scattered across the year. So you can only compare these 20 colonies to these 20 colonies, and these 20 colonies to these 20 colonies.
And
which doesn't give you in that window, and that doesn't give you a lot to select from. But the fear was they raise brood all year long in Hawaii. Those colonies are coming out brood continuously.
Yeah.
So are the inseminations going to last for a year? Can you go on a yearly cycle?
Yeah.
And so that was another one of the concerns. So there's a lot of different reasons why we decided to do what we did. But in the end, we got good VSH there. But when we brought the bees over for field trials, honey production was not good.
was lacking. Yeah.
It was lacking.
Well, at least you got the trait stuck in there, though. You can always kind of work with it from there, you know?
Yeah, yeah. So and that seemed to be consistent. I mean, we did one beekeeper worked with us all 3 years, and then we had the last year, which I think was in 2020. We had 5 different beekeepers that had completely different. One was in Michigan. I mean, they all went to almonds, of course.
Yeah.
But, one was stationed in Michigan, one was in like Washington, one was in California, Dakotas, all over the place. And it was pretty much the same thing, even though the Hilo bees made more honey in Michigan than the bee, or than the control. We use, commercial Italians as our controls, so that the controls were standard in all these. But, they actually made in Michigan, but nowhere else.
Yeah,
I mean,
really?
Yeah. Oh, in some places like in California, you couldn't judge because, the average was like 5 pounds,
Yeah, yeah.
Well, it's 5 pounds and 5 pounds, what does that mean?
Do you think
they pulled a frame of honey, woo!
Right.
But they weren't, and the thing is that like in California, they weren't interested in honey production. They don't make honey there. They want them to brood up. I think it was a smaller queen producer. So, he wants bees to raise queens. That's where his money comes from, that and almonds. So, they don't make as much honey. I don't care because I don't make honey anyways.
Right.
Other people, it's like, yes, that's my main, besides almonds, that's my main crop. And I need them to make honey.
Mm-hmm.
So that's where we're working on now with the Hilo bee. Is we've changed everything up. It's completely different now, which is why before I was just kind of, Bob Danka was overseeing this because he's a scientist.
Mm-hmm.
I'm the technician. So, I collected the semen to ship. That was my job. I collect semen to ship. When we first initiated it, but then we were like, okay, we need to change things up because this is all those SDIs and the resistance was getting so high that what we were doing was having colonies that produced mites, shaking mites out of those bees, and then infecting all these nukes with like 200 or 500 mites each to test. And that's very, very labor intensive.
Oh yeah.
And of course we had grants to help start this program, but eventually those go away and it's got to be sustainable for the beekeeper himself, even with selling the queens. So, the closed population, Page laid off closed population breeding program is the most commercially economical way to breed the bees.
The standard.
So, yeah, I mean, it's been proven, Sue has proven that it works. So, and then there's been others that have done it. Joe Latshaw in Ohio, he does the same thing with an Italian and a Carniolan population. And we've been doing it with Pauline. So it's like, okay, we can do this there if the queens can last for a year.
Mm-hmm.
And so what we ended up doing was, and this of course went on in 2020, the worst time that we could be doing this type of stuff. We mixed it all up where we basically separated the population into 2 subpopulations. So we had basically what we were trying to do was like a spring and a fall population. And in the fall, or in one of those, we were going to import Pauline semen.
Okay.
Just to give it, we've been, I've still been breeding Pauline and it's just there for research. We might as well put it to some use. So we were basically going to have like we call it a Hilo subpopulation and a Hilo Pauline subpopulation.
I like it.
And so it's like every other year for the HP population, subpopulation, we'll import semen from Pauline. And eventually we'll stop doing that. But currently right now, it's like, that's what we're doing. And then the other, the HH, the Hilo subpopulation, is just the Hilo bee continuous on. Well, this involves not inseminating 20 queens every month. This involves inseminating at least, our goal was to get around 200 queens laying. So that meant we had to inseminate at least 300 queens twice a year.
Right.
And with inseminations, if you don't have that type of practice, and they had 3 people in Hilo that can do these inseminations.
Mm-hmm.
They had 3 people that were trained to different degrees on how to do this. They had different experience levels. But when you're doing 5 to 10 queens yourself once a month, that semen collection is a lot different than having to collect semen for 300 queens.
Way different.
I saw your video. It was like, this is the part that I hate about insemination. Collecting semen. I'm actually different. I actually don't mind that part.
Yeah, that's good.
But when people start inseminating, they either pick up one or the other really fast. They either pick up the semen collection really fast, or they pick up the queen insemination, and they struggle with the opposite.
Yep.
Very few people pick them both up with no problem. Of course, there's just some people that can't do either one.
Either. That's probably most people.
Yeah. Well, the people that actually would come to like Sue's classes and stuff, they're kind of vetted to a degree, but they, it requires working under a microscope, the fine detail that you have to do under a microscope. Some people just can't do that. But typically you have one can do one or the other really well first off, and they have to work at the other.
Mm-hmm.
I was the semen collection.
That was easy for you?
That was easy. The queen insemination was hard.
Yep.
And then the other thing that helped with that is when I was at Ohio State, Sue was very protective of her bees. Mm-hmm. So it wasn't like, oh, you want to, oh yeah, go ahead and, inseminate some queens for me to put out here. She was like in control of that, especially when you've only been doing it for like a year.
Right. I don't blame her. I
don't either. I'm the same way.
Yeah.
I'm the same way. I'm like, oh, well, this person here, can, they collected semen 5 years ago, learned how to do it. They can help you. No, they can't.
Right. We don't
not until they've done it and I see what they've done. Then I may give them approval,
Make sure it's not straight mucus.
Yeah. So we would sit down like the second, actually just continuous on after I learned how to do insemination. So it'd be like, okay, we're inseminating queens today. I collected drones. So get your instrument set up. So we get everything set up, and she would come, go grab 4 cages of drones. Mm-hmm. Okay, here's your 2 cages. Here's my 2 cages. And then we're collecting semen. And of course, she's done like that.
Quick, yeah.
Yeah, she's done. She goes out, gets some queens, and she inseminates the queens. And then she's like, how far are you along? Oh, you almost got 100 microliters.
Yeah.
Okay, here you go. Collect more.
Right. Yeah, I'll take that.
So the only way I got to inseminate queens to get practice, I mean, I had my own bees and I did practice on my own.
Yeah.
But to really get, we did a lot more at the lab at Ohio State. But the only way was I had to get that semen collection fast enough that I was starting on a queen before she finished her queen. And if I could get that, then I get to inseminate those queens.
That's awesome.
So I got really good at semen collection.
Yeah.
But in Hilo, with the people there, they didn't, they only had to collect 30, 50 microliters tops once a month, not 800.
Big difference.
So, and then of course I'm trying to train them through Zoom, all this stuff, and we're changing everything up completely. I mean, we had Arista Bee Research. The guy started that, Bart Jan.
Yep.
He was the one that was hired to oversee the breeding in Hilo.
Is that how you pronounce his name? Bart Jan?
Bart Jan.
Okay.
Yep.
Excellent.
Yeah.
I'd
So yeah,
seen
he
it
was,
written. I just hadn't heard it
yeah,
spoken yet.
yeah. So he managed that, and he ran it a lot like how they would do it in Europe, because that's
Mhm.
what he knew. And of course, he's doing the same thing in Europe.
Are
I
you,
mean,
is the difference confined drone flight cages versus catching free flying like Sue
it's
does? Or
it's confined drone cages. It's this queen is mated to drones from this queen. It's line breeding
okay,
basically.
okay.
Um, and a lot of times the drone sources would be F1 queens that were open mated.
Mm-hmm.
But it would be, okay, well, we are going to do inseminations. Okay, we got this queen, we're going to mate her to this queen. And so they go have to put drone brood out, bring it back, put it in a colony, have a kind of flight box on top so that they could kind of fly and escape. And,
Can they poop in those or not at
yeah,
all?
they can, but they
Yeah,
still, to me, it seemed like they were always kind of unhealthy.
yeah,
Um, they didn't yield as much semen, or
yeah,
I mean, I always just say I need 100 microliters of semen, get me 200 drones.
yep,
I mean, it's 50% is the nutrition rate of either the drones are dying before I can get to use them.
yep.
They yield no semen. They contaminate. It flips over and hits my finger or hits
Oh
them
yeah,
or
good shit. Mm-hmm.
whatever. That's the way it is.
Yep.
So they had all of that type of stuff, which is what they do in Europe, which works great there because what they do there is they have like cooperatives.
Mm-hmm.
There's no like, oh, this is my bee and stuff. This is the Buckfast bee, and we have this breeder, this breeder, this breeder, this breeder, who all take theirs to an island to mate with drones from somebody else, or
With the cooperative.
a joint of them. So it's a cooperative, and that's what he's doing now. I mean, he's got a VSH Buckfast, a VSH Carniolan, and I think he's been working on a VSH German black bee.
Oh, cool. That's pretty neat.
So I mean, it's really expanded. He's got a lot going on there, and the reason, one of the reasons why he left Hilo was because he was just so busy.
Yeah, too
I
much
mean,
going on.
he's maintaining, trying to manage all this stuff there, and they were doing the same thing, SDIs, introducing mites, and they would do the same thing. They'd have a big old let's look at brood party, and they would have
Yeah,
10 people show up, and they'd all be there looking under a microscope trying to
that's
find
what I
mites.
did. Like, I've got rodeo tickets. Who wants to go? It worked, or I guess they just thought it'd be cool to hang out at my house. They kept showing up, so I guess it was
Yeah.
somewhat fun.
You didn't run them off.
Right. Hey, speaking of, I don't want to squirrel too much because I really like where you're going there. There was something that I'd thought of. I kind of ran it by Sue. She didn't say a whole lot about it, but with the concept of SDIs, I was thinking of a variant of that. Like, instead of a single drone from a certain queen, if you caught drones only from that queen, so they're not genetically identical, but there's some recombination with her DNA. But if you did like a single colony insemination to where you could give them 10 microliters, you would still have a lot less diversity than if you just caught, like you would Page Laidlaw. But if you gave them 10 microliters to where it wouldn't be as big a deal on the longevity, instead of them lasting 6 months, maybe you'd get another year out of them or whatever. But I don't know what your thoughts would be on that. Is there too much variation, you think, with that recombination, or you think that might be useful in a breeding program?
I mean, it can be, all of these different techniques can be used in the proper way,
depending on your goal.
Yeah, depending on the goal and what
Yeah.
what you have, an idea for, and what you want. That's basically how they were doing it in Hilo. Was they would have this queen would mate to drones from this colony, and this.
Oh, they were, okay. So it was kind of single colony inseminations.
Yeah, sometimes it was drones from these two, but those two colonies were sister queens.
Okay. Yeah. So they should
I
be
mean, because
genetically very similar.
yeah, so I mean, because if you think about it, you're not really mating her to that queen, you're mating her to that queen mother.
Yep.
And so that's how they looked at it, was this was a good queen, here's her daughters. They would select the best, the ones that looked the best off of those, and then they would use those as drone source colonies.
Yeah.
So um, it does narrow your genetics.
Where you're not sifting through so many subfamilies.
Yeah, because you've only got one. You got different subfamilies, but the daughters in there are all half sisters,
Yep.
or super sisters or full sisters.
Right. Yeah.
Is what they are. So they're either 50% related or 75% related to each other. So you have that. So it can be useful if it can be. You can run a whole breeding program just using that.
Yeah.
But it's a lot of work, and you need a lot of colonies, because you constantly are going, okay, this queen, no, I can't use that because they're too close related. So I got to use drones from here. And she didn't have drones now. So now where can I?
Yeah, exactly. Welcome to bee breeding.
Yeah. But for, say, you want to lock down the trade a little more, or there's something particular that you're looking at, and maybe you do a small subset like that to really look at something specific with that group, that you may be able then to incorporate those back into the main population.
Yeah.
That wouldn't be a bad thing. Because I will say that in Hilo, what we're seeing now, granted, this is only only starting the second year of actually doing this type, this moving to the closed population breeding system. And actually in 2020, it really didn't start till the fall of 2020.
Yeah.
They kind of maintained a little bit because we were hoping, we'll open back up and I can travel out there. And,
it'll only be 2 weeks.
Yeah. No, I can't. Okay. Well, I will ship you semen and you can inseminate.
Yeah. Did that work out pretty good?
It did. But there again, there's a big difference when you are trying to introduce 20 queens versus 200.
Big time.
The best thing, which they didn't use, they used them for other purposes, but I used them. I don't know if you've ever heard of Scalvini cages. So I had one around here somewhere. This is one I'm trying to 3D print. It's not, I had a real one somewhere, but it's maybe it has to be in front of me. There we go. So it's this cage that has worker cells in the bottom, and then it has a lid that closes, and then a little cork plug that plugs in there. Now I made this cage so that the workers couldn't get in, but the normal ones are queen excluder material.
Okay. Yep. Where they can pass through it.
Yeah, they can pass through it, and the queen's stuck in there. So in Europe, what they use these for is to produce a broodless state in the bees.
Okay.
So they can treat for oxalic acid.
Okay.
So they put the queen in here, and this is narrow enough that the cells are deep enough that she'll lay, but they're not full. So the brood can never develop. So they just
cannibalize it.
They'll cannibalize it, and she'll just keep laying.
Okay.
So she's maintaining laying. They're interacting with her, so they aren't trying to supersede her
because
in the colony.
Yep.
So that they can make that broodless period, so they can treat. Well, one fall I was like, I typically had made nukes up, 5-frame nukes, introduced the queens. Either had really good nukes and had an excluder, one of those dial things where I could exclude the queen from flying.
Yep.
Yeah, or staple excluder on there, or push in cage. And I was doing some inseminations in the fall, and I was gonna, they had to overwinter, so I was just gonna try to introduce them into full-size colonies that I dequeened. I mean, I made them, like a full deep, something that can overwinter, but not
Yeah.
overly huge. And I was like, man, these things are not bee tight. I'm gonna have to put them underneath a push in, but with all those bees, they they'll
Underneath.
chew her out
Oh,
before
yeah.
she starts laying. And so what
Yeah.
am I gonna do? And I was like, I wonder if a queen will lay in those. I mean, well, we know they lay in it, but I'm wondering if an inseminated queen will start laying in one of those
Yeah.
because it's plastic. I mean, it's all plastic.
Hmm.
So I tried it and it worked.
Nice.
And I'm like, this is great because I can put her in there and she can be in there, and then I can go back and check and see if she's laying. And if she's laying, I can release her. If she's not, I don't have to.
Yeah.
Because I know you've probably done the same thing I have.
Oh, yeah.
I mean, I introduce them with a JZBZ cage.
Hmm.
I put her in there for like 3 or 4 days.
Yep.
I mean, I inseminate her. I leave her in the bank over the weekend, basically.
Hmm.
Gas her a second time. I put her in JZBZ cage and I let her keep her in there for like 3 or 4 days. And then I move her to the Scalvini cage. And usually by the second day, 90% of those queens are laying.
Really
Cages.
nice.
But you know what? If you don't do that, what ends up happening? She's running around in there, and you go to check. Well, I don't see any eggs. Well, I didn't see the queen. Did I miss her? Is she on the bottom? Did she escape? Is she dead? What's going on? And then you're like looking. Well, I didn't see her. Maybe I just missed her because she's still running around, acting very virgin-y like. Okay, I'll close it up. I'll come back here later. You might do that a couple times. So you're like, okay, she's definitely gone.
Yeah, something's up.
Yeah. This, I put her in there. Of course, you got to check for queen cells every time. I mean, that's just something to do with inseminated queen. But then when it's like, is she laying? By the time I put her in the JZBZ, usually I don't have to look for queen cells anymore because it's already been 4 days, since she was in there, when I tore queen cells down. So, there's already shouldn't be any queen cells left, but you can just literally go in. Nope, she's not laying. Put it back.
Done.
Or oh, she's laying. Let her out. Or she's dead. Okay.
Yeah.
I know.
But you don't have to search everywhere. You just have to look in there.
It's got little box. That's
I like that.
what she's in. And she most of the time she doesn't die in there. She typically ends up dying in the queen cage before you put her in the Scalvini. Every once in a while they'll die in there, but it's not as common. Usually they're dying earlier than they are when you put them in there. But it speeds things up so much because you can just go in there and be like, nope, laying. And I went in like 50% of the queens will be laying, around 50% will be laying within 24 hours.
Dang, that's awesome.
But I always go back there 2 days because 90% of them are laying in 2 days, and then I don't have to go back a second time.
Again. Yeah.
And so I was like, hey, we're going to do this for introduction. And they've never done it in Hilo. And so they were like, oh, okay, well, that's kind of weird. Does it, well, we always did it this way. But, I'm trying, these are the ways that I have found that are more economical and faster,
That's big.
And that's what we're looking for.
Yep.
So, and I don't know where I started when we started the story, but
it's been good though. I've enjoyed.
I don't know what point I was originally trying to make, but now
So how would you spell that? Salvini, Salvini?
It's SC.
Okay.
Let's see. Stupid computer.
Yeah, that's pretty cool. I've never seen those before.
Yeah, it's called It's spelled Scalvini.
Salvini. Nice.
So I've got a little write-up that I made up because I've had some people ask about it.
Yeah.
I keep meaning to put it on our website, but and there again, I probably have to go through all these channels.
All the chains,
to make sure. But I can send that to you.
That'd be awesome.
So you can see what I'm talking about, because I mean, honestly, it's great. I even asked Better Bee, I was like, hey, can you start carrying these,
Mm-hmm.
because I mean, for us with the federal government, it's a more of a hassle to buy stuff from overseas, which is the only way you can buy it,
Yep.
than it is to buy it within country.
Makes
We
sense.
can buy them. I mean, I've got 100 or so of them, and they're not cheap. I think they're like around $12 a
Each.
piece, but
Are they from Denmark or somewhere or somewhere?
something like that. Yeah, I think
Yeah.
there's a company from Italy, from Denmark that sells them. I mean,
Okay. I didn't know
there's
if like,
several companies.
have you ever ordered from Sweenty Bee Supply in Denmark? They've got, I think the only thing I've ordered from them, I don't know if they have something like that or not, but they're like Man Lake, and some of them would have numbered discs, but it was all red, and I
Yeah.
wanted color coded ones, and I ended up getting them from Sweenty. But they had all
Okay.
kinds of like micro gas setups for instrumental insemination and instruments and just stuff you don't see in USB supply.
Yeah. Yeah. Well, insemination is a lot more common over there.
Over in Europe.
Because I think like Poland, I think most of their queens are mated with insemination.
That's insane. Wow.
Yeah, they've
Yeah, I
got.
know some of the like Sue has her method or reasoning why she does something, and then there's like scientific literature to back up why she does it and when she does it. And I know like Woike had a plethora of stuff. It's like that guy just put out pure gold constantly, and I think he's maybe still around. I talked with John Keefus not too long ago, and he mentioned Woike. But man, that guy was a genius. He had so much good stuff.
Yeah. Yeah, that's some of the stuff you do, it's just it's personal. Like this works for me. I mean, it's like that. You ask 10 beekeepers how to do something, you get 100 different answers, because
Exactly. Yeah.
I mean, this may work for you, it may not, but it does for me. But those Scalvini cages for me just speed things up tremendously.
It makes perfect sense, really.
I mean, I don't have to look. She's confined. She can't get out.
Yep.
Unless I don't lock the door.
Yeah.
I mean, that's the only way she gets out.
Operator error.
Yeah.
Yeah.
Which happens anyways. I mean.
Oh yeah, it's gonna happen. At least it does in my operation. I'd like to think I'm not alone.
dropped a cage, walk into the truck, didn't realize it, and find a dead queen. There's all types of stuff like that happen. But they work great, and we used them in Hilo. So last year I was able to actually travel. They were allowed mission critical work to go there, and we were able to say, hey, look, this Hilo bee, this is a big project of a lab, and if I don't go out there, it's gonna slowly die off, because they need to be able to do this.
What was your
because
boss?
they were gonna inseminate, catch semen, and all that stuff on their own. It's like they just don't have the practice and the skill level yet to do that volume.
Yeah.
Plus, we had to do so we do 300 queens for the breeding population, in hopes that you get 200 laying.
Oh yeah.
So that's the goal, 200 laying. So we do 300, because you always lose. I mean, that's the other part that's hard about inseminating queens, is getting them laying.
Yep. It's true.
You gotta know how to raise queens. You gotta know how to take care of the drones. And each one of these are a difficult level. How to inseminate the queen and how to get her introduced.
Yep.
That's just as hard as learning the insemination.
And then the oddities of nature, maybe they want to supersede her for some reason, or it seems like you're always going to have some level of oddity. So that was a good call. Do 300 and account for it.
Yeah, and the goal of having 200. But then the beekeeper also, we started selling Hilo queens to the public under a request. So you had to request, and you had to basically agree to these certain criteria. Like you're not going to propagate these and sell them on your own. You're not going to buy them and resell them type
Right.
thing.
Yep.
You know, you understand this is a work in progress. This is not the final product. You know, so, you know, please don't go, you know, saying, well, these bees suck.
They're horrible.
Yeah,
It
you know,
get
just,
worse.
and, you know, okay, yeah, here's my, like, and we released, like when we did those field trials, we released the data on that. I mean, we said, look, mite resistance is great.
Yep.
Overwintering is great. Honey production is terrible. I think it's like
Yeah.
50%.
Yep.
You know, we're not hiding anything. Like this is the way it is. This
That's
is
the best
where
way
we
to
know
do it.
we have to work on this,
Mm-hmm.
and which is what we're doing. One way is switching to these subpopulations, because now this population is all raised at the same time, and we can judge the honey production of that population throughout the year, which in Hawaii is hard too, because they don't pull honey once a year or twice a year. They're pulling honey continuously throughout the year because it's tropical beekeeping.
Yeah, they're just constantly moving.
It's expensive to get the equipment in. The colonies are kept in a single deep with an excluder, and then, you know, one or two mediums on
Mhm.
top, and they're always, you know, honey's always being plugged in the brood nest, or pollen's a big one over there.
Really?
Just getting plugged out with pollen. They're just removing frames of pollen.
Man,
Yeah. And so,
that's
so
cool.
they have to keep track every time they go out there. Oh, I pulled one frame of honey. I pulled 2 frames of honey. I pulled a medium box of honey, whatever it happens to be. So it's not this whole, oh, I wait till August and I take them off and I mark them and I go weigh them and boom, there you go. I got it. It's,
Mhm.
you know, all these little things throughout the year, because
Pieces
they
added
look at these,
together.
colonies every 3 weeks. They look
Okay.
at them. And then every 12 weeks, they do what we call a detailed check, where they actually go through them, verify the queen, and then take a varroa sample.
Mm.
So every time the other checks are more just like beekeeping checks. Is the queen right? You know, do they need room? Is everything okay?
Basic stuff.
Yeah.
Yeah.
But they still, you know, it's easy to take a population count. That didn't take long. So I take a population count. You know, they don't go through to find the queen, but if they find her, we saw her, you know, that type of stuff. Just,
Mhm.
but it's not a requirement. And even on the detailed, I'm like, if you go through twice, you don't find the queen, don't waste your time finding the queen.
Yeah.
Yeah. We got other times that we can find her. What is important is when we say, yes, this will be a breeder, then you have to make sure that that's
Yeah.
definitely
Yeah. Right. For
but
sure.
you know, so we're doing that to get the honey production. So we're getting honey production data off of them now. And then what we also did, which is kind of mind-boggling and scary at the same time, is, you know, this beekeeper has David Thomas. He has 7,000 colonies on that side of the island. So last year, and he went for, I think it ended up being, it was over a year. I think it was like 18 months without treatment
Oh man.
on the entire population, and then
Yeah.
then he had to treat.
Mhm.
But what he did was, twice that year, last year, 2 times, he took mite samples of every single
Oh my
colony,
gosh! Out of all 7,000.
all 7,000. And
Wow.
then what we did was we pulled in the best 100.
That's awesome.
And of course, you know, we have no idea whether, I mean, some of the queen, like if the queens were queens because they came from the mating yards, they were marked. But if they were a cell put in, they weren't marked.
Sure.
So if there's an unmarked queen, you don't know whether that was the original queen that's put in there, or a super seizure, or,
Mm-hmm.
you know, or super seizure of an original one, because a marked one, because you don't know whether it was marked or not anyway. And,
Yeah.
you don't keep that detailed information on 7,000 colonies. You keep, this is how many colonies they got in the yard
Mm-hmm.
type deal.
Yep.
So we brought them all in, went through, marked all the queens if they weren't marked, and then just started watching them. And these were 0 or like 0.1% mites
That's
when
awesome.
they did these washes.
Yeah.
So we pulled in 2 different sets, because one was done in July and one was done in like August, September, somewhere around there. So we had these 2 different ones, and we basically went through and marked all the queens, and then we started watching them the way that we do the subpopulations that we work on, and basically said, if anything happens, if they are mean, if they have terrible brood, if there's any reason to reject them, they're rejected.
DQ.
Just like that. And the goal was, if we get 1 or 2 that we can add into the population or add in as a drone source for the population, that's great.
Yeah.
But they also, when they brought these in, they also went out and made sure that it was a good looking colony too. And so they just didn't say, oh, these are low mice, we're bringing them in no matter what they look like. They went out there and looked and said, okay, this one's not very strong. That one's not, we're not taking that one. This one's really strong, we're taking that one. So we've got like these several things where we're trying to improve the honey production in different ways. Along with those field trials, we collect the best queens from those field trials on the mainland. I collect the semen from them and ship it off, and we inseminate queens with those too.
That's awesome, man. I'm pretty excited about that program. It sounds like it's got to go places. You guys have got enough experience, and it sounds like you keep tightening it up and getting better and better too, which that's, should be the goal of any breeding program.
Yeah. And every time I go out there, we end up doing more queens. Because we do all this in 2 weeks. I don't know if I said that. So we inseminate 5. Our goal this time is to inseminate 500 queens.
Oh, man.
300
awesome.
for the breeding population, 150 for the beekeeper.
Yep.
Because he's selling queens and he needs something.
Breeders.
He watches them on his own and does some mite samples, and then selects out the best ones that he's going to graft from. Because we don't want to give him, he can have ours after we're done, but ours are a year old after we're done.
Yeah.
And they don't last very long in that heavy duty commercial queen rearing environment. So this is like the best of both worlds. He gets 150 that he goes and he selects his best X that he wants for breeders based upon mite levels, how good the colony is, whatever more beekeeping traits they has, but it's the same stock that we're looking at.
Yeah.
And then this year, what we're trying to do is, which we've always talked about doing, it's just, the pandemic, all that stuff. The lab does research on these bees also. So we're trying to do like 50 queens to bring back to the lab so we can use them for research, but also as kind of a backup, because all that stock is on the one side of the island in Hawaii.
If something happens, or
a hurricane could come through, volcano could erupt and potentially lose it. Then that way we've got something we could send back to redo.
Yeah.
Eventually the goal would be to freeze semen.
I was going to say, do you guys do any cryo or anything?
No, we bought the equipment back in 2019. I think it was in the fall of 2019 for me to learn how to do the cryo, but then I wasn't allowed to go anywhere to learn it.
Where do you go for that? I mean, I know there's a conference at WSU coming up. I'm going to go to it in July.
Yeah, I'm hoping to be able to get to that.
Really? That'd be awesome. Maybe we could catch up and have dinner or something while we're there.
Yeah. I'm, still got to see how the travel situation is and stuff at that time. But,
do you think it'll still be goofy? Are you talking about on the employer side?
On the USDA side.
Yeah. Do you think they'll loosen up by then, maybe? Or
I'm hoping so.
Yeah.
I mean, we're just now at on the 28th of March is when everybody's allowed to come back into the lab to work.
Okay. Well, that's progress.
So even right now, not everybody is back at the lab to work. So, so that's the first step. And this research, like for, research purposes for that, like I go to, they've been very generous in allowing those if you can
Good.
prove that it's a necessity. So,
Yeah.
but the big thing that they hold out for is meetings. They're like, meetings, you don't have to,
Yeah. It's
meetings
not
are
business.
still, don't even ask to go to a meeting because we're not going to
Yeah.
let you.
Yep.
So hopefully by then it'll be loosened up. But yeah, so you've got Brandon there in
Mhm.
Washington, and I think he's got a postdoc or a student, a grad student or something like that knows how to do it. Because he offered for me to come up last year. He offered for me to go out there and learn it, or
That'd be awesome.
he could have sent his student, or I'm not sure whether it was a student or a postdoc, but whoever it was, to come to the lab and to teach me at the lab,
That's
which we
pretty cool.
still were not allowed to do. So
Really?
so it was
Yeah.
like, and it's not mission critical that we
Exactly.
learned that, so it wasn't, we couldn't really get anything approved. The other one is Arun, and I can't remember his last name. He's at the USDA lab in Fargo.
Okay.
So he developed cryopreservation for semen around the same time Brandon was doing his. So they both were working on it at the same time. I think Brandon was a little earlier,
Yeah.
obviously, but there's Fargo is has a big cryo lab type thing there. So he worked on the semen, and he got that down. And we actually did a little test. Brandon and Arun came down to the lab in 2018 or 17 or something like that, and I inseminated queens from frozen semen. I collected semen. They both froze it for a day, and then we inseminated queens just to see which one was better. Was there any difference? And
was it like buffer solutions or something that they're using, or?
it's the solutions that they use.
Yeah.
I mean, Brandon won, and there again, because I'm doing all the beekeeping stuff, I don't get to be like involved in some of this stuff as much as would be nice.
Yeah.
But Brandon, the one thing that Brandon does use is he uses egg yolk in his,
Oh, interesting.
which is very common in cryopreservation of semen.
Mm-hmm.
It's kind of the traditional. Arun uses is all chemicals.
Okay. Yep.
So whatever it's just chemicals that he substitutes for the egg yolk. I mean,
Yeah.
the formulations might be a little different. But when we did it, there's basically no difference in the end result.
Yeah.
Yeah. You dilute the semen by like 50% when you freeze it. So they're in little straws of like 20 microliters.
Okay.
You inseminate 5 queens with each straw.
Do you give her the whole? Do you give her 10 or?
You give her 5 microliters.
5.
So
Okay.
5 queens per straw. So each queen gets 4 microliters, I guess it is. So
Okay.
It's about 20.
Yeah.
In a straw. So it's about 4 microliters a piece. And what you end up happening is one queen doesn't do anything, whether she doesn't lay, they don't accept her. One out of the 4 or out of the 5 is gone.
Yeah.
One of them lays great. It's all worker cell. That's great. The other 3 are degrees between the 2. Not quite all drone layer, but not 100%.
Yeah.
Brood. So 100% worker brood. So they all seem to end up like that. And Brandon took some back to Washington to look at, and I kept some here, and our results were the same. So I kept mine going to see how the longevity was, but I was also doing other things, so
yeah,
it wasn't that detailed. Just something like, it wasn't something that was part of the experiment. It's more just like, I
Yeah.
wonder.
Casual observation. Yeah.
Yeah, well, let me see. Let's see what this. I do a lot of that. I'm just like, hey, let me.
I love that. That's how you learn stuff,
Yeah,
That's why I've and somebody will go, "This is, gospel." I'm like,
yeah. The only
like what parts make that gospel? You know what I mean? Is it still true if this changes, or I don't know, which if you like variables, beekeeping's where it's
yeah,
at because it's innumerable variables.
yeah. I always like when I see online, I see somebody like offering a insemination class,
Mm.
and they're like, we teach this, we're really good at this. We have over 95% success.
Yeah.
That's like that's 95% must be in the percent that you inseminate and they don't die in the
Die,
bone.
right. Yeah.
Because
Low bar.
when you introduce them, if you get 90%, you're doing really,
Yeah,
really good.
exactly. Yeah, there's so
I
many
mean,
factors because even if the queen's good, if the bees aren't having it, and maybe you weren't cautious enough, or I don't know, there's
well,
just an innumerable number of things that can go wrong, it seems
yeah.
like.
Well, I've had colonies where I made, split down 2 colonies for queens,
Hmm.
and the splits from this colony, boom, accepted her, no problem. The other one, it's like
No.
killer. Next one,
Nope.
killer,
Yeah.
killer. It's like they had to be no brood whatsoever. Then they're
Yep.
like, okay, fine, we'll take the queen.
Yeah. Since it's
Or,
either death or her, I guess we'll take her.
yeah, but
Yeah.
no, we still got sealed brood, so we're not going to give up.
Yeah, I
Maybe
know.
one will be a queen.
Right. Yeah, I got to where, because I would hold Sue's like, hold them, it's good to hold them in their own nuke. Well, I wasn't paying attention. I would make it up and put her in there and they would feed her and everything's great. And I would inseminate her and I would just go turn her loose because, she would say, like, if she can run around, the sperm migration's best.
Mm-hmm.
I would just turn her loose thinking they got to know her and everything's great, but I didn't pay attention. They had queen cells down there and I don't know how many I got murdered that way. But if I went and cut the cells and held her for a little bit to where they knew they were screwed, then they were more accepting of her. Maybe not 100% by any means, but your odds were better.
Yeah.
But
Well, I found that if I make the nukes up with very little young brood,
okay,
I can have open, I can have larvae,
but
if
not
they're
queen rearing age.
yeah.
Yep.
And some is fine, but the worst ones are always here's a frame of eggs or just emerged, an entire frame of just emerged, hatched eggs.
They
That's
love
like,
to make queen
they're
cells.
just like queen cells, sequence.
Yeah.
But if
throw her out.
Yeah. and they'll just do that. They'll be like, okay, well, they're not even, we're just starting the queen cells. Yeah, get rid of that. We're not even gonna think about it.
It's
So
true.
I have found I try to make mine with all sealed brood or the order larvae. Sometimes, if there's a few eggs, I'm not, it's beekeeping. I'm not that picky
Right.
on stuff.
Yeah.
But if it's the whole frame of, well, there's eggs on this side and newly hatched larvae on that side, I'm not taking it.
Yeah. Too big a risk.
Yeah.
Have you ever done any like packages or bulk bees? It seems to me like that would work well. I mean, I've done where there's no brood, like you said, that they seem to, if they're healthy, they seem to take them extremely well then. Like, because I got some breeders from Harbo a couple years ago, and I don't know what I was expecting, but it's, I'm the same way probably. Like, hey, are you ready to take your queens? Well, he just kind of let me know that they were already on the way, and I didn't have time to like come up with a good plan. And so I just found a decent sized colony and took every single brood frame out of there and with the queen and just took them off. So there's a ton of bees, which there's field bees in there too, but just a mass amount of bees and plenty of stores. Took her, they were just happy as could be to get her, which I held her for a little while. I didn't just candy release her or anything, but And that I thought that was kind of similar to bulk bees or a package because there's no brood or anything, just
Yeah,
lots of bees and resources.
yeah.
But
I've never done. I mean, we've done packages or bulk bees into packages for experiments,
okay,
but those queens that we put in there were already laying inseminated queens.
gotcha. Okay,
But I've never done like never the queen's never
not yet
been
learned.
laying. Yeah.
Mhm. Yeah,
Um,
I started selling some of those, which I think the jury's still out. Which I'm going to add the flush, the extra step on it, and see because some of them they would email me back and be like, "She's in there." It wasn't like my bees killed her, but she's not laying. So I would just send him another one because it was something
yeah.
wrong with the insemination or something wrong with the queen. But
Hmm.
it's so much cheaper if you don't have to make up all the nucleus colonies and put her in there. And, I would just have healthy queens. And I got the idea from John Harbaugh.
Yeah.
Inseminate them and then ship them, which they're cheaper. There's more risk involved, but they're cheaper, and people love cheaper. Well,
Oh
I
yeah.
say people in general, some don't. Some would rather pay more and make sure she's laying. But
Yeah.
yeah,
Yeah.
I know he sent out a questionnaire. I need to ask him about it. I'm in his breeding group and have been since it started, his little breeding cooperative.
Hmm.
And yeah, he sent out a survey afterwards if you had bought untested, and I wondered if he was kind of getting a percentage of what to expect, how many failed and how many made it. I'd like to talk to him about that and see what he found out because
Yeah.
I'm kind of doing the same thing, which I'm not selling any. I usually sell a few that are laying, like I call them just select layers. But this year I'm supposed to graduate in May with
Oh,
a master's in entomology, and I didn't want to bite off too much, and I didn't want to. I always bite off too
okay.
much. I didn't want to be able to not make it happen, and
Yeah.
overcommit. So I'm just doing virgin queens and like freshly inseminated breeders and a few breeder nukes. But
Okay.
anyway,
Yeah. I mean, it's I don't know. I very rarely have shipped off queens to somebody that I didn't have the queens laying to begin with,
hmm
just because of how difficult it can be.
agreed.
Um,
Yep.
but there again, the more I do, the more experience I get, and but there again, there's still variables. I mean, I
Constantly.
expect. I mean, I'm happy if 75 or 70% of the queens I get that I inseminate get successfully introduced in laying.
Yep. That's a good goal. Yep.
And to me, laying is after the first brood cycle emerges,
Mm.
because you can get them laying and they'll die. Some of them will die or be killed before
Or be
that first brood
superseded.
cycle. Yep.
Yeah.
But if they make it until after that first brood cycle,
Generally, they're
usually
good.
you don't have to worry about it. They
Yep.
usually don't raise any cells on her, and you don't really have to worry about it.
hmm
And I've even gotten to the point where I just maybe it's just because I work for the government and I don't have to worry about nickel and diamond
Yeah.
every little thing to make
Yeah.
ends meet. But
yeah.
I don't even go in and look for queen cells anymore. I mean, I look for them like, oh, I'm introducing all the queen cells now, and she's in the Scalvini. All she's laying. Any more queen cells? No, okay. And then I don't worry
Yeah. She's good.
about it
Yeah.
anymore. If they
Mhm.
raise queen cells and supersede her, they usually are doing it within that first. I always go back after 30 days from when she starts laying. And if they superseded her, I'm like, oh well, it probably was.
Moving
I could have
on.
probably prevented it from a queen, going and looking, but
Take
when you're
another trip, more digging. Yeah, exactly.
yeah, and when you're doing, you know, 200, I usually for the pollen, I usually inseminate around 250 queens.
That's awesome.
So, am I really going to go dig around in 250 queens
That's a
for
lot of
that?
digging.
And that doesn't mention the experiments that are going on,
Yeah,
that are inseminations, all the beekeeping that has to be done, because I mean, you know, I might be working research for bees, but the bees still need the same beekeeping that needs to be done anywhere else.
absolutely.
Not to
Yeah.
worry about, like I'm going to Hawaii, leaving for Hawaii to Hilo for tomorrow
Yep.
for the round inseminations. We're going to inseminate 500 queens in the next 2 weeks.
It's insane.
And we'll more than likely we'll have those done by like Monday or Tuesday of next week.
That is cool.
The goal is to get them inseminated and to at least have them all in nukes, introduced into nukes to various degrees.
Yeah.
I mean, some of them might be, laying, like on the last day, we might be going through looking at Scalvinis and releasing laying queens, but then other ones are going to be moved to Scalvini, different stages. And, I always leave a handful back in the queen bank for, oh, this one died. Okay, well, we'll just
go grab another one.
I'll grab another one and put in there. But that's the goal. But I'm going to be gone for 2 weeks, and this is the end of March, April here in Baton Rouge. Things are flowing right now.
Yeah.
I mean, there's not a honey flow or anything, but nectar's coming in, pollen's going. The bees have been building up like crazy. The first drones, the colonies now are full of drones. And I'm like, I'm going to be gone for 2 weeks. So I'm out there, trying to get the colonies supered. So I don't, so that they
Give
don't
them
have
a little room.
Doing that. I've got 140 mating nukes out there that, because we, in Mississippi, goes like this. So we have, like, by the lab, we have one of these peninsulas that has all Pauline colonies around it. And the lab uses 80 colonies or 80 Pauline queens for research purposes.
Nice.
Not breeding purposes, just to research.
Yeah.
And so we tried to set up this like naturally mating area for Pauline. So, I'm trying to get those all established and get the cells in so that they can be mating while I'm gone. So when I come back, I can harvest the queens. And oh, it's April now. Now you got to do all the queen rearing for this and that, and do the inseminations, and
it's a lot.
It's a lot. And we're still working. When I got to the lab, Jeff Harris was there, and he knew how to do insemination, and I did. And then when he left, it was just me.
You. Yeah.
and you'd have technicians it'd be like, hey, or the scientists would say, hey, we want them to learn insemination.
Yeah.
Okay, I can teach them insemination. And then okay, that's great. They put it in their their plan. This year they're going to learn instrumental
development plan.
Yeah. And they'll be like, okay, so you're going to teach him, right? Yeah. Okay. Well, he's got half a day today.
Can you teach him?
Can you teach him?
Yeah.
That doesn't mean I have half a day today.
Right. Yeah.
But he has half a day. Okay. Well, here, let me spend a half hour. Here's how, put the instrument together.
Mm-hmm.
Okay, take it apart, put it, take it apart, put it, here's how you do it.
Basics. Yeah.
Take it apart. Now you put it together.
Yeah.
You start off like that, and okay, and then you get so far, whatever that day day of training is, or half a day or an hour, or whatever it is. And then it is like 2 months later, hey, he's got another half a day.
Yeah.
Okay, we'll get the instrument together. I don't remember how to put the instrument together.
Yeah, it's been 2 months, right? Yeah.
So nobody could ever, because when you learn insemination, you gotta have a couple days where that's all you think about. Yep. And then you have to practice regularly after that.
Exactly. That's what freaked me out is I learned from Sue, and I think it was a July or August whenever I went. And I usually raise fall queens, in August, but I didn't get to try it. So it was basically, the following spring, which, thank God I remembered most of it, but you're still rocky and you don't know what you're doing. And, you kind of, it's like you were saying, just the experience level, it just takes a while to iron a bunch of stuff out.
And the thing is that you're rocky, like, every spring when you start off, you're rocky again.
Yeah, because you haven't done it for so long.
So frustrating. You're like, I know how to do this.
Yep.
Why isn't it working?
Yep.
Why can't I get that tip in? Why am I collecting mucus? Whatever the thing is, it's like not working. And then after like the first round, then it's like all the muscle memory
easier.
comes back and it's like,
Mhm.
boom, okay.
Yep.
But that first day, like when I came into the lab, and it was like that first day in spring, it'd be like, I would shut the door to the lab and it'd be nobody bother
No
me,
distractions. Yeah.
because I'm going to be frustrated because
Yep.
something's not going to be working right. And it could be the instrument, it could be my inseminations, whatever. There's
Yeah.
always something. And I tell everybody that that's just the way it is. Maybe after you do enough, like now for me, I really don't have that, but I also
It's easier.
inseminate. I just inseminated queens last week.
Yeah. I haven't
I
done
had to
anything
get some
last
I had to
spring.
get some STIs done for an experiment, so I inseminated like 6 queens last week.
Awesome.
And and I will be inseminating queens in October
Yeah.
and all year through. So and I'm gonna I probably inseminate 800 queens in a year.
That's awesome. I don't do that, man.
I know it's a lot.
It is a
But
lot.
after enough years of doing that, it does
It minimizes.
it minimizes it, and you really don't have. I mean, I did have a little bit of issue this year because I was doing single drone inseminations,
Mm-hmm.
and I'm trying to pack stuff up to go to Hawaii. So I was trying to do it
Distracted a
the
bit.
minimal amount of stuff I had to have out.
Yeah.
Usually I set it up, and there's my station, and it stays like that all year long.
Mm-hmm.
And so this is like, well, let me just.
Oh, you had to get ready to take it with you.
Yeah.
Oh,
So I didn't want to like
gosh.
set everything up, because I'm just gonna have to tear it all down. So I was like, okay,
Yeah.
let me just, the bare minimum.
Bare minimum.
And,
Yep.
and of course I'm like there, and I'm like, okay. And then I forgot, because if you don't know, when you do SDIs, you have to put in, usually in the tip, you put in 2 air bubbles before you start collecting semen.
Okay. Yep.
Yep. That way, when it gets into the capillary, it merges into one big one, and usually, hopefully, if everything goes right.
You don't dilute the heck out of it.
Yeah, hopefully they don't get little small bubbles, and then you just have this, gradient going.
Yeah, I've done that. Yeah, I know what
I
you're
have
talking
to.
about.
I mean, when I went out to Hawaii the first time, and we were, okay, this is what we're doing and how we're doing it, and I've been doing this for so long, and I've been doing it by myself.
Mm-hmm.
You do this stuff by yourself. It's not like you sit there.
Oh yeah.
Usually in a group is not, having 3 people inseminating at the same time is unusual in the same
Very.
area. So I do it by myself. So I'm like, we're playing this out, and I'm like, yeah, we can get this done and this done and this done and this done. They're like, what?
Yeah.
I'm like, yeah, I mean, we need 1,000 microliters of semen. Well, I can collect 500 to 800, microliters in a day.
Mhm.
500 normal, 800 if I really try. So,
Yeah.
you guys should be able to get the other 500. They're like, excuse me.
Yeah, they're like, no, we're not going to be able to do that.
And so I had to realize that, ooh, wait a minute.
Different.
I've got a lot more experience,
Mm-hmm.
and I'm a lot faster. I didn't realize how much faster I am, because you don't usually talk about this. When you're talking about inseminations, you usually don't talk about how much you do or how fast you do.
Yeah, I don't know that I
like
ever
that.
really have. Yeah.
Or even if you pay attention. I mean, usually it's just like, I'm just trying to get the queen. I got 20 queens I want to inseminate. Today it may take me 4 hours to do those, and it might take me 2 days the next time. I mean, who knows?
Mm-hmm.
But when you do this constantly and you're always trying to fit it in, it's like, okay, see, I can collect 100 microliters in 45 minutes. If the drones are good, if they're bad, it takes about an hour. So I can get this done, and you're doing all that type of stuff. Or I can inseminate this many queens in an hour, and all that. And so I had to like go there and be like, okay, wait a minute. And then I start talking to people about this. So, people from Kona came over. They do inseminations with their carniolan, and so like Kelly, his son came over, and we were talking, and he's like, yeah, we did 300 queens, and it took us, took me and this other girl about however long it was, a month or something like that to get them done. And I'm like, he's like, yeah, because we can get this, we do about this an hour, a day. And I'm like, oh. And then I start talking to other people, and it's like, okay, so like for the most part, it's probably like Sue, Joe Latchall, and myself, and maybe a small
that are
handful of
at
others
that.
that are at that level. And
Yeah,
everybody else
exactly.
is like, and that could just be that's where they're comfortable at. It could just be, and then what I told the
And
people in
skill
Hawaii,
level too.
yeah.
Everybody's gonna have a different skill level in it.
And I told the group in Hawaii. I mean, there's 3 of them, and they're great, but they also what they caught on to was the queen insemination, not the semen collection.
Yeah,
that was their thing.
that was their thing.
And so, but I also had to say, okay, I'm going to be doing, and we got a lot to do, so I'm going to be working. I'm here to answer questions, because that always, I mean, you probably would like that. Crap, I got a mucus plug. How can I get that out?
Yeah.
Or I can't get this tip in the queen just right. What am I, can you come look at this and tell me what's wrong? So I was like, I'm here to answer any questions.
Mm-hmm.
I'm going to be working as hard as I can to do what I can. But for you guys, what I want is quality.
Yes.
Don't worry about not being fast enough. Don't, quality is what we're working on. The speed comes later.
You're exactly right.
You can do the speed, and if you don't have the quality, you never really get the quality back.
Yeah, if you got 5 tubes that are half mucus, you're going to spend more time trying to clear plugs and, than you are inseminating queens. It's just not worth it.
Yeah. And then they humbled me last year. So I went and we were doing the inseminations, and I wanted to see how good they were.
Mm-hmm.
And I mean, they collect the semen, you can sit there, watch them, and okay, they look like they're doing it right. And, they inseminate the queens okay, and the queens lay, but really how good? So, and they've had issues. So it's like, okay, where are the issues at? Is it on the insemination or the semen collection? So what we did was we all collected semen, and then I took all of their semen and inseminated the queens, and they took my semen and inseminated queens.
Yep.
That way we could narrow down where the issue was, and we're on of them. And really there wasn't any issue. I think the only issue we had was Donald was his light source, which we had already corrected from the previous time. Because you buy all these microscopes and they come with these LED lights.
Yeah.
They're blue.
Mm-hmm.
So they have a bluish tint to them. The semen and the mucus look great to me. Look gray.
Yeah.
Hard to distinguish. You need a light source that is shining at 3000 K, which is a little more on the yellow side.
Okay.
And you can tell the difference. So
interesting.
And we use regular fiber optic lights for the microscopes at the lab, so we never had that issue at the lab. But there, I was like, I can't tell the difference.
Yeah, it's
like,
hard to...
oh well, we bought this light source from whoever's making the Chalet instruments now. He made a light source that's supposed to be the correct wavelength for collecting semen. But I looked at it and I couldn't. This was one of the girls that was inseminating, Juliana, and she's like, I just can't tell the difference.
Yeah.
I don't see any difference. So, when it's, a little more awkward and stuff, so I didn't use it. I was like, okay, well, let me see. Like, oh my gosh, this is amazing. I can see now. So I used it, and everybody else said they were fine. So, I used that one there. And then at the end, Donald, I was like, okay, well, we can buy more of these if anybody sees a difference. So she was like, well, let me see. And she was like, no, I still don't see a difference. And I take it to Melissa, the other girl, and she's like, nah, I really don't see a difference.
Really?
Yep. I mean, now Juliana, her thing was she actually wears her glasses at the microscope, and she had blue filter glasses on for the computer.
Okay.
So I thought Melissa, the other girl, did too, but she didn't. She just couldn't tell a difference. No, I really don't see a difference. I took it over to Donald, said, look at this, and he was like, oh my gosh, I can tell the difference.
Yeah.
So he
I
uses,
never thought about that because I bet all mine's LED. Like I've just got a cheaper AM scope and it's got the LED halo underneath it. And then
Mhm.
I've got like articulating, you can flip it up and it's magnified and it's a LED halo, like for putting
yeah,
on, for doing VSH assays or if I'm putting, discs on the queen, which I don't usually use magnification, but it kind of helps a little bit. But I never even
yeah,
thought about the light wavelength. I wonder if I should
yeah. There,
potentially look at that
if you
too.
go on Amazon, you can look for LED light sources at 3000K,
Okay.
and they have these. I mean, they look like the fiber optics with the flexible arm
Yep.
type thing. I mean, they're a little bigger than what the end of the fiber optic is because that's where the LED is. And at least the ones that I found typically are made to screw into something.
Okay. Mm-hmm.
I don't have one here because I got it packed, but that's what I use in Hawaii.
So something around 300K. That's
Yeah,
the yellow range you were talking
yeah,
about.
and it's a little bigger, but it's small enough that it fits nicely under the around the scope, especially if you have a boom stand.
Yep, I
Um,
do.
it doesn't work great for me at the lab, but I got a regular lights at the lab because I use the microscope that John Harbaugh used to do inseminations.
That's awesome.
So it's just a
That's,
little, Bosch alone with the regular
yeah.
dissecting scope that's probably was there since 1970.
Yeah.
But
Yeah.
everybody else has got these big fancy
Oh
scopes,
yeah.
and I'm like,
Yeah.
that's
Cameras plugged into
yeah,
them. Yeah.
yeah. But it made a big difference. So I think that for what because that was like at the end of the trip because we were looking at buying some if they helped.
Mm-hmm.
And since it just helped him, I was like, okay, well he can use that. I'll bring something of my own from the lab. And so when we did all this mixing of switching semen and all that stuff to see who was having issues, nobody really had any issues.
Yeah.
So that was good. The semen looked good, but as I said, they humbled me because since they struggle at collecting semen, when it gets down to crunch time, I collect all the semen and just hand it off to them to do the queens.
Yeah.
Just like Sue Kobe again. I'm just like semen.
Yeah.
But they would sit there and they'd be inseminating queens. They'd be like, Garrett, yeah, I got a mucus plug.
Really?
Semen.
Yeah. It happens to the
Oh, that's like I said. It happens. I mean, that's
It
just,
does. And it doesn't
especially
take a lot either. A
no,
little bit can cause a problem in that super fine tip.
yeah.
It's
Well,
a,
and my tips are even worse. The ones that I make
yeah. Are
because
they?
I polish them really
Oh,
well.
okay.
They're like the tip comes up and then it's like smooth
Yep.
on it. So I always bought tips until I came to the lab and they have a tip puller, and so okay, well I'll make my own tips.
That's
And
awesome.
I made tips, comparing them to what I got from Joe Latshaw and Sue Kobe, and okay, yeah, those are right size, all that stuff looks good. I inseminated a batch of queens in the fall, and they all died.
What
I'm like,
was it?
what happened? Contamination? I'm going, you're going through your head. What was blah, 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 blah. I was cleaning the instrument up, and I was cleaning that tip, and the tip literally sliced right into my finger.
Was it
I didn't
broke?
polish. No, it wasn't broke, but it wasn't polished enough that it just literally, I'm like, well, if it did that to my skin on the inside
What's
of the
it doing to
queen,
that queen?
it just,
Yeah.
no wonder I was getting it in really easy because it's just like slicing
Slicing
right
right
there.
Yeah.
No matter where I put it. Wow, that worked.
Slice right through the valve fold and go right to the
Yeah.
oven.
So I ended up like overly polishing them, which is great because they literally just go, slide in because I have a.
That's awesome. I'd never see this stuff I've never thought about. Like I just buy mine from Sue, but I've also accidentally, I don't know if it was while I was cleaning it, but I noticed that I broke the end of it. I don't know if I dropped it or what happened, but just stuff you don't think about and really minute things like that can cause an issue. And like the last batch of queens I did last year, not good. Like, I don't know that I got any of them out of it. So it makes me wonder, like, and I'm just thinking contamination, but now it's got me wondering maybe it was the tip or something, you know?
Yeah.
Hmm.
Yeah. But because those things, because I have them so smooth, the mucus
Yeah.
also just
Goes right in.
and you got it to where it's like in pieces and stuff, and it could just slide right in, and then it always ends up staying at one spot,
Yeah.
and then a little bit more comes in, and it like finds that mucus and just makes it bigger and bigger and bigger until it's an issue. And I tell them, and I tell everybody, one, have a question about insemination that I don't mind. Ask whatever you want, because whatever it did or whatever you screwed up on, I've already done it.
Yeah. Mm-hmm.
Trust me, I have done just about everything you could think of.
Yeah.
So
that's the best way to learn.
But so also when you have a mucus plug, this is what you do. If you can't get it like this, this is what you do. So when you have these things, ask me, or I can't get the air bubble out of the syringe. Why is that? I've done this and done this. It's not working. What am I missing? And and it's just sitting there fiddling with stuff until you find it. And I was like, well, have you tried this? What? No, I never thought of that.
I haven't thought of that. Yeah.
And it's like, yeah, I didn't either till it happened to me. And
you had to figure your way out of it.
Yeah, eventually you take everything apart to the smallest pieces and put it all back together again and figure out what it was. So, but yeah, so it's like all that happens.
Oh yeah, there's so much that can go wrong. Like one of my buddies was down here. I don't know if we VSH tested or what, but I was going to inseminate some queens. He's like, "Oh, do you mind if I videotape you?" I'm like, I don't mind. And first one, you get one of those queens that won't. Like, I don't know what, if I didn't get around the valve, something was going on. She wouldn't take it. And I started getting frustrated. And finally, I'm like, just turn that off. delete that footage. We don't want that getting out. Yeah, no, it was just. And then, like, I did some insemination for Zach Lamas, and this was like a cold start, like I hadn't done any. And so, yeah, it was like problems. And I ran out of gas because I have a big tank and I never think about refilling it. I need to get it back up. But then I took a break, got a gas refilled, had a breather, got something to eat, and then like a sewing machine. And it was easy. And he's like, "Oh my gosh, you're done with that already?" Like, once you get moving, if everything works right, it does not take that long to knock out a batch of queens, But
have you tried giving them 2 injections of semen, like 2 small doses?
No, I've just done the 110 microliter dose. Is pretty much what I've stuck with. Because I know, like, back in the day, they used to do 2 subsequent, and it was a smaller, like 3 or 4 microliters, something like that. Is there an advantage to that, you think, or?
I've been messing around with it, well, I guess for about 6 years now, since 2016, I think.
Okay.
And it was a little bit by accident. I was trying to develop my own type of syringe because,
nice.
like the Harbo syringe that Sue sells uses the Gilmont. The actual syringe part is a Gilmont syringe, and those can be iffy on quality. Sometimes you can like go like this and the semen doesn't move, or and Sue had them like specially make ones to her design. But there at the lab they just buy them. They don't buy them from Sue because they can order directly from the manufacturer, from the company. So they just one of the scientific companies, so they just buy them. And so you always have to go through and find out which one is what, because they went from boring the plastic to make them to mold injection.
Okay.
So the quality just wasn't as good.
Wasn't the same.
Yeah. And so I was like, and I use a Latshall instrument for inseminations.
Okay.
And so I had his syringe, which is basically a Harbo syringe. He just made the syringe part himself, which is just a tube, with not a needle on the end. He just used one of the rubber connectors, or he used a silicone connector that fit into the hole, and then he just had a piece that screwed on with a metal threaded rod that
Okay.
screwed in with a O-ring,
Mm-hmm.
on it. And that's like really, really smooth.
Nice.
And I was like, well, I would like to design one that was a little fuller. Well, and then he changed the syringe, and he actually has one that collects like 200 or 300 microliters at a time. He has
Nice.
a large capacity, which actually fits up in the instrument, and the whole barrel. It doesn't use a capillary. The tip just fits into a
Okay.
barrel, and it collects to a glass tube, really, and then
Okay.
it just collects it and fills it up there. So you do have to collect quite a bit in there because it's wasteful, because there's no such thing as like an air bubble, and the tip has to be full of diluent
Mm-hmm.
to start. So if your tip holds 20 microliters, there's 20 microliters of saline at the very end. So if you're collecting 100 and then you push it out, you're and that of course dilutes, you're not really getting as much. But when I do the inseminations, I sit down, I collect like 800 microliters at a time. I collect semen one day, inseminate the queens the next day, do some work, collect drones, that type of stuff, on and on and on. So I was like, well, if I can get one that collects more than 300, that would be great. And so I was trying to make one, and that bigger volume and all this stuff. And I was like, okay, well, this will work. And I measured out, calculated the volume, how many turns was it for, how many microliters did you get out of a turn and all that stuff. And so I'm inseminating queens with, this works great. Inseminating queens. Then I do something, I decided to recalibrate it to make sure, and I was completely off.
Really?
So I'd given all these queens, like, because I was like, wow, this semen's like lasting a long time. This is amazing. And here I was only giving them like 2 microliters of semen instead of 8.
Whoops.
And I'm like, oh crap. So I went back and recalculated. So I went back and recalculated. And so what I ended up doing was a small batch of queens that got 2 microliters 3 times.
Yeah.
And then a group that got like 2 microliters one time, and then 4 microliters the next time.
Okay.
And then I did like the standard 10.
Mm-hmm.
I was like, oh well. And it's so behold that the ones with the multiple insemination seem to last longer.
Really?
Like at the fall of the following year, and basically any old queen is getting dequeened for a new inseminated queen.
Mm-hmm.
Well, the only ones that were left that were not, that the only ones left that were inseminated were the ones that had the 2 or 3 smaller doses.
Interesting.
Of 6 microliters of semen versus, I think I gave them 8.
Huh. What do you think? Do you think they're getting more semen migration, or what do you think?
That's the theory. I mean, that's what the literature says, is that smaller doses, more semen, a higher percentage migrates to the spermatheca than if one large dose. So I've played around with it. I have no real hard data to back it up.
Sure.
It's just something
observation.
that I'm doing. I would do, one year I did half the queens one way, half the, 2 doses, I think it was 5 microliter doses each, and then 1 10 microliter dose. One year I did all double inseminations, 5 microliters both times.
Is it 5 one day, 5 the next day, or do you skip a day, or?
Yeah, typically it was like 5 one day and 5 the next.
5 the next,
And because I was just, curious and looking at this stuff, and I was like, well, looking at the first year's information, it looks like this is good. So I did like half and half, and then when I'm doing the next year, I don't have the previous year's data yet because it's only a year.
Mm-hmm.
So I'm like, well, it was looking good that year, so I'm going to do half and half. I'm going to do all of them double inseminations this year.
Yeah.
it looks like it's going to be good. And I did, and it was fine. And then the next year, started the pandemic, and I'm like, okay, I don't have time to deal with this. That's just one full dose.
Yeah.
But what I noticed is, longevity aside, I really have no comparison, except for that first year's worth of data, because the second year, it didn't work out to where I could really check anything, because we were dequeening. We needed colonies, so if they weren't used for breeding purposes, they got dequeened. So that's just the way it is. But you tend to, and it really varied, you tend to lose more of the double inseminations. And I think that's just because of the handling.
That makes
your
sense.
handler, you're putting a tip in twice, it's a higher risk.
But if you get them in, they potentially might last a bit longer.
But there again, I'm thinking you got to know what you're doing to risk
going back.
Try and go back in twice.
Yeah. Because I think they used to do that a lot, didn't they? Like, if I remember some of the earlier stuff that I've read, I think that that was kind of the routine, was to do 2 smaller doses.
Yeah, because they mimicked, they tried to mimic nature as much as possible. Yeah. So it was, the queens were inseminated in the afternoon. They would typically be mating. They take several mating flights, so we're going to give them, 2 small doses, and all that, and then eventually change just 1 single dose. And
there was some, I was thinking, it's been a few years, but I know that, which I just use straight CO2, but I thought Sue had something that had come out that said like that some people maybe had advantages if they used a blend, like an air blend that had X amount of CO2 in it or something. Have you heard anything on that, or do you just use straight CO2 as well?
I use straight CO2.
That's what I've always done.
I put them in basically put the queens in a Ziploc bag.
Yeah.
Push as much air out as I can.
I do
don't
too.
get it all.
Yeah.
Fill it up, let it expand, wait till the queens aren't moving anymore. Turn off the CO2, seal the Ziploc bag up, let it sit for 3 minutes.
Yep.
Open it up, dump them out, and let them wake up, and then release them.
That's what I'm doing too. Shabu said he was doing like 5 minutes or even a little bit better. I wonder, at what point does it kill them? Does it take quite a while?
I think it takes them a while, because there's been times I put a timer on for 3 minutes, because there's been times when I put them in there, let me go do this, and then I get sidetracked, and then it's like, oh, the queen, and then you're like, and you're dumping them, and they're like not moving. Wake up! They're not moving.
Come on now, shock paddles. But you think it takes quite a while for them to actually?
It takes quite a while, because I've heard of people giving 5 minutes, 7 minutes, 10 minutes. I know that in Hilo, their original thing was, I think it was 7 minutes.
Okay. Yep.
And it was 7 minutes for both gases.
Okay.
So you inseminated the queen, and you can get her inseminated in 2 minutes. You had to sit there for 5 minutes until you could do the next queen.
Yeah.
And I don't do that. It's the queen's in, inseminated out. Queen's in, inseminated out.
That's what I've always done too.
Usually she's in there for under 2 minutes.
Yep. Mm-hmm.
Unless something is not going right. But
I have a technical question. You may not know, but you know how, like, whenever you go to grab the stinger, there's 2, like, I don't know what they are. It like a sting sheath or something. It's not a sheath, but it's like, you know what I mean? There's 2 of them, and it like holds the stinger in between it. Sometimes you have to get them out of the way. What's the technical name for those? Do you know?
I don't know.
I made up some terrible name a while back, and like I couldn't think of what to call it exactly. And I don't think I've ever seen it on a anatomy diagram or anything.
Yeah,
Sorry, no,
I don't
I don't
know.
mean to
No, 'cause, like,
sidetrack us
I'm
there.
like
Just
thinking like, no, I don't know if
yeah,
I know what
I
that's
don't know what
called.
those are.
Yeah. Huh. Yeah. Have you ever run into split stings?
Oh yeah.
Yeah.
Hmm.
Those are hard. When I learned
What
insemination,
causes that?
I
Just
have
the
no
sheath?
idea.
Because I think on a stinger, or at least on a worker bee, there's two halves.
And the same with the queen. There's two halves that
So maybe it was damaged or something, or maybe
they,
she.
yeah. I've seen them where they've been like split into like five
Oh wow! See, I haven't
different
seen that.
different sections.
Dang.
I've always like made notes thinking I'm gonna go back and like raise daughters off of her and check to see if
See if it's
her
genetic.
daughter, yeah, and I
Yeah.
never do. I
Yeah.
mean, it's
Yeah. That would be
it's like,
me.
oh, I need to do this. So I write a note, and then it's like,
Yeah.
then I don't, and it's like, oh
I'm the
man, I
same
should.
way. I say I'm kind of an ADD kid. I mean, not to where I can't do things, and I'm always working on something. And but yeah, my attention sometimes goes from one cool thing to the next.
Yeah, yeah. If um, eye mutation pops up, then I'm a
Have you
goner for
seen
that too. I'm like, say it, we're gonna
have you
inseminate that. And
have you seen red-eyed drones before?
I've seen red eye, white eye, cream, chartreuse Benson.
What? Are you serious?
Yeah, I
I've
actually got
seen white. That's the only thing I've seen so far is white. I've
yeah,
seen pictures of other stuff, but I've not seen it in person.
yeah. Well, the chartreuse Benson actually was um, sent to me from Harbo when I was in Ohio.
Yeah.
I was curious, and they used at the lab they used to maintain all that stuff.
Okay.
Before genetics are the way they are. All
Mm,
the genomic stuff. They maintain mutations to use for experiments.
yeah.
And so he sent me a queen that was homozygous. She wasn't homozygous, but she had the trait in her. So, her offspring would throw 50%,
That's
and
pretty
so
cool.
but I had to watch it because then like all of a sudden it's like half my colonies are the side
Right.
mutation because I'm just like, oh, this is cool. Let me trying to get like pure homozygous,
Yeah.
not workers because then they kind of can fly a little bit because they darken up, they can kind of see. But it was
But
like, no, I just try to get more and more.
yeah.
It's
Yeah.
like, oh, but yeah, I see those every once in a while. You see them, but I got to be like there because somebody will say, oh yeah, by the way, when I was out, I saw this. Did you catch the drone?
Yeah.
Oh,
No,
no.
it flew off. Right.
Not even just for like to preserve it, to store it, to say here it is.
Yeah.
No, but yeah, I'm into the weird stuff like that. I had a conversation with Wade Law one time because I was like getting into this would have been mid 90s, and I was really getting into like the mutations. I love the cordovan mutation.
They're awesome looking.
The
Yeah.
Pauline, I've almost got it to it's 100% cordovan now.
Really?
Yeah,
That's awesome.
it's not yet, and it's taken me, you know, 10 years
Yeah.
to get to this point because, you know, I don't want the color. I'm not selecting for the color. It's kind of like an addition. Like if a
It's a
queen is throwing wild type and cordovan drones, I'll pick the cordovan off
yeah. Mhm.
first. But then if I run out of those and I need the other
Grab
ones,
the others.
I
Yeah.
use them.
Yeah.
At least there initially. Now, it's enough that almost every queen is throwing cordovan drones.
That's cool.
Last
Those
year I was
things look awesome.
able to inseminate with 100% cordovan drones for the
That's
entire
too cool.
population. But the previous generation was also queens that I did those double inseminations on,
Okay. Yeah.
and I didn't, because of the pandemic stuff, I didn't have to kill them all off to make room for stuff last year. So I had a bunch go through winter, and they still, look good. I reevaluate every single thing every year.
Hmm.
So even if they were a breeder last year, doesn't mean they're a breeder this year.
Yeah.
I mean, I used to do that. I used to be like, she's a breeder, I'm going,
For life.
yeah, she
Yeah.
looks bad her second year because, she's dwindling down and dying. But,
Right.
now I'm just like, no, I'm just going to reevaluate them every year. New mite washes, the whole deal.
Start fresh.
And yeah, and some of them look good and some of them made the breeder cut. I mean, I have some that population, all that stuff is good. And I have four mite washes over 2 years with under 1%.
Nice. Yeah.
Every single colony has a drone comb all from the get-go. So she could raise drones all she wanted. And so that population is not all, even the drone sources were not all cordovan. So
Sure.
Yeah.
I
was like, oh, this next year it's all going to be cordovan because, half, all the drones are cordovan and now I can do all drones cordovan again. And then I was like, pick these. And I'm like, well, there goes that because most of them are throwing cordovan drones, at least half, but I know of one that is not at all. She's throwing no cordovan, but it's one of those that
she's too good.
Yeah, she's just too good to say no. And I'm like, I really want to say no to you, but when they produce, 3 deeps of honey and have low mite washes like that, and and they're gentle and the brood looks great. It's like, how can you
can't kick her to the curb.
It's like, man.
Yeah, maybe next year.
Maybe can I just raise queens off of you and not take your drones? No, that's awesome. Not fair. But yeah, I mean, all that stuff. I had like I said, I had a talk with Laidlaw when I was getting into it, and he sent me like all of his. This was back when the internet was just getting started, so you had to ask for reprints of stuff. So he sent me all of his reprints that he had on mutations, and in the email that he sent to say, hey, I'm sending all these. His last sentence was, "The mutants will rise again."
That is really cool though, studying mutations. That's awesome.
Yeah, it is.
Man,
but you don't need to anymore.
What's that?
You don't need to study mutations anymore.
No,
you can just pull the DNA and look at the entire genome.
Scan
no problem. Yep.
Man, that's crazy. I'm still I haven't I haven't really dove into that a whole lot. I know, I'm not a geneticist by any means. I know that they can do that. I don't know exactly how they do it. I'm sure it's computer running through millions of sets of alleles and whatnot.
Yeah,
it's crazy that we can even do that now.
Yeah. I mean, I understand enough. I mean, I don't know the process by any stretch, and I don't claim to. I'm a beekeeper first, a technician second.
Yeah.
And I'm a field technician. So, I mean, I can work under a scope, dissecting brood or, dissecting bee, that type of stuff. But, if you want me to go into a lab and like start mixing chemicals to extract DNA, I'm going to have to be trained from the very beginning on what to do and how to do it. I'd have no idea how to do any of that stuff. And then running it and then looking at the computer program that analyzes it all. Yeah. And we got people that do that. So that's fine. We got a great guy that does that. But he doesn't really like to be out in the bees.
Yeah. It takes all kinds.
He's not a bee person. So, okay, well, you do that. I'll do the bees.
And it works.
Yep.
It kind of makes me wonder. I know like Allison McAfee had written a while ago about like genomics and gene marker assisted selection and stuff. I just wonder if that'll be more of a thing going forward. I don't know if it's totally practical yet, like if you can send a sample and go, yes, this has all the VSH alleles or whatever.
We're trying. And the biggest issue that happened with that is this whole, when we found out the recombination rate of honeybees.
Okay, that threw a wrench in the works.
Yeah, because we had a scientist who, this was late, or what is it? 2010, 2015, in that range. And she had did this work for like on corn and stuff.
Okay.
Marker selection and all that stuff. And so she got hired into the lab, and she was like, yeah, this will be no problem. And so we did the testing, the phenotyping, she ran the stuff, and like, okay, well, you have potential markers, and then you have to like test it. So then you give her samples and she says yay or nay, and then you either have the phenotype data to back it up, or you have to go get that, depending on the situation. But you had that data to verify that it was working, and it never did.
Really? Because of the recombination?
Because of the recombination.
Interesting.
And once that came out, she had actually moved on to something, to go back up north to her family. And that's when the high recombination rate was coming out, and she's like, that's it. That's why. She says it's going to be, you're going to have to look at more stuff to be able to do this, because
wow.
If I understand, like I said, I'm no expert in this.
Yes, I'm not.
But if I understand how it worked, was you don't find the gene or the alleles that are responsible for the trait. You find something that's close on the chromosome to that carries with it.
Okay.
But when you have all this recombination,
it's
got to be a lot closer, or you got to find the exact alleles.
That's what I was thinking. It almost has to be the exact gene or the exact set of genes. So it may even be multiple, not just one, but like with VSH, like they said, they believe it to be multiple. You'd have to nail.
It's multiple, and it's additive.
Yeah.
So you can have this and that, and it works okay, but you add this in, and it works really good, but you take one little thing out, and it goes back to, it's very confusing.
Yeah, indeed. I think we'll probably figure more out too about it. Like, that kind of leads into one question I had for a while. Is I thought that, like if they would do freeze killed brood, like they said, it may or may not predict mite resistance. It will predict hygiene, like chalk brood and stuff, but it may not predict VSH per se, or specific mite hygiene. But then
it doesn't. I have a paper on that.
Well, the thing, really, okay. Well, I think was it Marla that did that? Was it a Spivak paper or?
Well, mine was different.
You did one, okay.
Yeah, which was spurred by Marla and Sue. I got in California for a different reason, and I got permission to go like visit queen producers. And so I was like in the Davis area, and Marla Spivak happened to be out there doing the free scale brood test on breeders. And so we met up, and Marla Spivak made the comment that she felt VSH was just regular hygiene on OCD.
With additional alleles, maybe?
Well, she was just thinking it was like OCD. Like, she stopped her free scale brood. She stopped at, because they cleaned it out in 24 hours.
Yeah.
It was hygienic. And she's just saying that with what we did, she thought that, and this was, at that time, her thought was that maybe we just pushed it a little further.
And do like 6 or 8 hours or something.
Yeah. And so we actually did an experiment where we looked at controls, VSH, and Minnesota hygienics. And we did the free scale brood, and we did a VSH assay. But the free scale brood, we literally would go in like every 2 hours.
And see how
and just take a picture. So we didn't have it out very long, but we just take it out, take a picture, and put it back.
Yeah.
And to see, because my thought was, well, you can do that, and you can find a correlation that like in 2 hours or 4 hours, they remove this much, and that signifies VSH.
Yeah.
Then that'd be great, because what we've always been trying to do is find a faster and easier way to select for VSH.
Yes.
And it just so happened that it didn't work. I mean, we saw some that remove stuff out, but what we saw was basically VSHBs are hygienic, but hygienic really didn't show into VSH. Some were, some weren't. You know, when you looked at the removal rates and everything.
I had said, I made a statement that I thought to be true, and I don't remember if it was Jeff Harris, or somebody that would know, or I would think would know, said, no, that's not accurate, but it's been a while and I don't remember. The statement I said was hygienic or freeze killed brood may or may not be VSH, but VSH should always be hygienic. But somebody said, no, I don't think that's right. But that kind of threw me for a loop, because I was thinking VSH is a hygiene mechanism, which is why they changed it from SMR to VSH. And I know I've never seen a single cell of chalk brood since I started using VSH, and I used to see chalk brood all the time, you know, and it completely eradicated it. So that, I don't know, do you think he read what I said wrong? Like, do you think that's an accurate statement, or do we not know fully yet, you think?
I don't think we fully know. I'm trying to remember that experiment that we did. For some reason, I'm thinking we had a colony that was VSH that did not do as well on the freeze-killed brood
So
test.
that may be where
But
it came from then.
It might have just been one. I can't remember. I mean, that was, you know, whatever, 2010 or something like that. And,
Yeah.
you know,
Do you think there would be any utility? Like, I've got a 50-liter liquid nitrogen Dewar with like an easy fill on it. I bought that, and then I got hung up on VSH, and that may have been around the time some of those studies came out that hygiene doesn't necessarily predict specific suppressed reproduction. And so I just switched to just using breeders exclusively and picking out of those. And then a couple of years ago, I started using Harbo's assay, but I haven't gone back and tested everything that tested extremely high with the liquid nitrogen. It's just been you know, it's one of those things like, I need to do that, and I've never done it. You know, but I'd like to, but I just haven't done it. But because there's somebody said, oh, so-and-so from Canada, which I know the guy, I've spoke with him because he's been asking me a lot of questions. He said he had VSH that didn't test high on hygiene, but I don't know that he has super high VSH stuff. So that was in the back of my mind too, because I know he was trying to get it, but there's kind of some misinformation or lack of information floating around. I think I won't say misinformation.
Yeah. And like I said, the level of VSH that can play a big part in it, because there's been, like in New Zealand and France, I think they were trying to work on stuff with VSH, and they were breeding their own, and they were seeing maybe different results than what we were, and it was because their level of VSH was nowhere near what we were at.
Yep.
And the same thing that can happen with you know, queen producers. I mean, you know, you have some, you know, and these are the smaller queen producers. Usually they buy breeders from VP Queens or, you know, Tom Glenn, you know, then there used to be Tom Glenn, and they buy them, and then they're like, oh, we got VSHBs, and it's like, you know, we tested that one time. We bought some from different people to see what their VSH levels were, and, you know, in the VSH level, you know, the lab was here, Tom Glenn was just below, and then everybody else was just kind of a gradient from, you know,
nothing,
just under Tom Glenn to nothing.
Right.
And when you start inquiring, it's like, well, yeah, I bought a VSH queen, you know, from Tom Glenn 4 years ago.
Yeah, and kept grafting out of her daughters. Their VSH.
Yeah.
Right? Yeah.
So it's like, yeah, it just continues on. It's like, no, that doesn't work like that.
Yeah. I've got ours to where last spring, all the breeders I tested in the first run were all 4s. It was all 100% VSH, and most of my F1s were. We found one that was like I guess you'd call it 75%. So they had one reproductive mite out of the sample. But of course I'm cheating. I'm using instrumental insemination, you know, whereas it's not, I'm taking the open mating aspect out, and I'm also unrelenting on the selection pressure too. But I'm trying to encourage other people to do some testing as well, just so, because I think that my thought behind it is, like you said, there's people selling VSH that
Mhm.
probably are 0%, or you know what I mean? Not what
Well,
I would
you'll lose
call
it
it
so
VSH.
fast.
Yep,
I mean,
exactly. But if they keep that selection pressure, you know, like if you teach them, and I know everybody's looking for easier ways to do it, and maybe like Kara's UBO, that spray. Have you heard about that?
yep,
UBO assay.
yep, I've
I'm
used
supposed
it.
to catch up with her next month, I think, and have a conversation. So she had some more data coming out that I was gonna talk to her about. But maybe if we can find better or quicker ways to where people actually start doing it, or even if it doesn't get easier, but all the cool kids are doing it and you're kind of getting left out, you know, you're not so
Yeah,
cool if you don't test for VSH. Maybe you could
yeah,
change the culture, and you know what I mean, to where more people are doing it.
yeah.
And then
Or
I
like the free scaled hygiene.
mm-hmm.
I mean, that was the same thing. I mean, it was like nobody did it. Nobody did it.
Yep,
Marlo Stiller went around and was like, "Here, we'll do it for
for
you,
you, yeah.
and show you." And then it got to be, you know, I mean, I'm not sure where it is now per se, but
Yeah,
you know, I think
know.
it's to the point if you don't test for it, you know, you're not one of the cool kids.
Yeah,
You know, that you're kind of
yeah,
left out. I mean,
yep. And that
granted,
changes the culture, and then if you change the culture, that changes the industry, you
yeah,
know. And I almost think VSH is going to see its heyday here, to
yeah,
where more people maybe start selecting for it. That's why I loved that you were working on the Hilo project too. You know, that they didn't just let it go. You know, after John Harbaugh left, that it still has its use and its merit, and you know, putting it in Paul lines and other
yeah.
programs like that, to where
And
hopefully
just
other people maybe start doing the same thing.
yeah, I mean, that's the goal is to, you know,
Hmm.
we want it to be used. Unfortunately, you know, I want to think that the amortized resistance is going to be the thing that causes people to,
Yeah,
unless they
strong
find another
arm.
chemical that they can just
Yep,
start using again. That's
yep,
cheap.
exactly. And
Um,
I talk, which luckily I took that, or I'm finishing up that entomology degree, and they just IPM'd me until I was just completely sick of it. But I'm glad they did, because it gave me, because I just came from a beekeeping perspective. I had some
mhm.
scientific training with insemination and breeding and selection, but like I didn't even think about things that way, you know. And then I think I can talk about it a bit better now and articulate it better, that, hey, you know, if you can find host resistance, that's like the cornerstone of an IPM. Because if you don't get any kind of host resistance, you might as well, I mean, you're going to be on the treadmill, of course.
Yeah,
If your beans or your rice or your honeybees have no zero resistance mechanism for primary pests, it's like good night. I mean, you're constantly having to knock it down, you know. And as if you can get certain levels of that, it's the only real way to not be completely
mhm.
reliant on chemical
Yeah,
minicides.
yeah. And there are like commercial beekeepers that are getting there.
Oh yeah,
You know,
I think
I
so
had
too.
a I had a talk with one at the American Honey Producers Association. A group of us sat down, and he was saying that, you know, because has used, you know, he was one of the cooperators that we did some of our Paul line stuff in. So
Oh cool.
you know, he got the genetics in there to varying degrees, um, and he might even work with, or he used to work with, if he doesn't now, VP Queens.
Okay, yep.
They kind of do the same thing. They're
Is
Paul
that the
line version
2.2?
2.2.
Yeah,
They
I've
basically
heard of
took what
those.
we did and tried to, I mean, they took regular VSH and Pauline Queens, but they tried to do the same thing as an additional form of selection for mites, which I think is great. You can test large numbers in a commercial operation, and then you can pull back the very best, and you can use the drones off of them
Oh yeah,
as drone sources. But
yep.
if they're open mated, you probably don't want to use the queens,
Right.
unless you are slowly introducing that in a way. But
Sure.
you know, he said that what he saw, what used to happen was in the fall, they had to work so fast to get their honey off that way
So
they get their mite treatments on so they didn't lose colonies that, it was nerve wracking, because of weather. It's like, we got to get this off, because like this time we have to
They're
have the treatments
done.
on or we lose colonies. So we
Yep.
have to. And he says now he still treats because, I mean, honestly, when it's your livelihood, you can't
Insurance
gamble.
policy.
Yeah. But he said the thing is now what he can do is they don't have to rush to get all that done. It's not a, live or die type situation. They can get it off. And if, oh, well, because of weather or this or that, we're 2, 3 weeks, 4 weeks late
Yeah.
getting our
No
treatment on,
biggie.
it's not a big deal. And to
Yep.
him, that's a game changer.
Absolutely.
I mean,
And
it's
it
less
is too.
stress for him. So
Yep.
he's like, he's like, would I love to be, no treatments whatsoever? Yeah, that'd be great. But, even if I can not have to, even if it gives me more flexibility
Yep.
in my treatments, is better than what I had before. So
Absolutely. And that's, like I said, with the IPM thing, it just reduces your reliance on that to where you're
Yeah.
not over the barrel immediately if, like you said,
Yeah.
weather moves in but
Yeah.
I think eventually if enough people are selecting for it and there's good commercial stocks available and you're working on it, there's going to be more come out. I'm sure you guys will have something great come out, and then maybe somebody else can do like Adam and kind of do their own program,
Yeah.
once that it gets more commercially available, I guess. And then people are washing out a bunch of ones and stuff late season. I think you're going to see people maybe even go to one treatment a year, or possibly, if it's just not even warranted from an IPM perspective, maybe only on bad years or something,
Yeah.
It's possible. I mean, I
Yeah.
think it's possible, but there's a
Well,
lot of
and
factors.
I think people have reduced their treatments overall anyways, or they're going with a
Softer.
a soft treatment in the fall and a hard treatment in the spring.
Yep.
When you can use oxalic acid in the fall and amitraz in the spring or something like
Right.
that. So that's something that that I think that things are slowly going in that direction. But the resistance trait, and VSH obviously is not the only trait for resistance either.
Yep, exactly.
I mean,
there's more out there. I think it's the best. I'm biased. I think
I do too. I think if you're looking at one trait, it seems to be the one that definitely can. The other ones reduce mites.
Yep.
But I don't know that you can really say that they can be treatment free.
Right.
And everything. Now, if you get multiple of those in there, like the Russian bee was bred just by looking at mite numbers. It was mite growth. It was, initially it was, we take a sample initially, and after I don't know how many months it was, they took another sample and they calculated the mite growth, and they bred from the ones that had the lowest mite growth. And so where, it eventually got to where they were getting negative mite growth in colonies.
Nice.
But they didn't check to see
what it was.
They didn't care.
Yeah.
Yeah. And, then when these things are coming out, you got VSH and you got grooming, and you have this VG where the brood actually is infested with virus or something and just dies on its own.
Is that apoptosis, if I pronounced it correctly?
I think so.
Social apoptosis.
Yeah. So, you got all these different things. And when they started looking at the Russian bees for this, it was like, oh, yeah, the Russian bees have VSH.
Yeah.
Oh, they got some of this, mite biting capabilities. Oh, they've got this, this and this. And, they have maybe not as high degrees as other stocks we have bred to, but they have multiple.
Yeah.
And it can keep it down. But, there again, it's still genetics and it's still bees. And, you've got that one that pops up every year or so that just, has the high mites or dies from varroa or something like that. But, overall, you can look at them and say, hey, they look pretty good.
Yep.
Low mites.
Yeah. And I think it's always going to be a bell curve. I think it's just moving the bell basically is the goal.
Yeah, I mean, it's going to have to be. I mean, you never go into a bee yard and see all the colonies producing the same amount of honey,
Right.
or all the colonies the same strength.
It's never
You
going to
go there, yeah, and it's like you have like a quarter of them that are doing great, and you have a quarter of them that are crap, and the rest of them are in the middle.
Yep.
And that's for like everything.
At
Quarter of
all
them are going to
times.
make excellent honey. Half of them make average, and then quarter of them make nothing.
Yep.
And
it's like a horse race. Sometimes you see two colonies. This one's way out ahead, and then you'll check them later, and this one's inched up into first place. It just seems like it's constantly moving.
Yeah, and for commercial beekeepers, they want those two extremes to be as small as possible,
right?
Because it makes breeding easier. But for breeding, I want the extreme. That's when we started doing this, working with the Hilo group closer, it was like, okay, well, we're gonna do these. We're looking at mite washes and stuff, and we're like, okay, well, this and that, or like Sue always pushed the 5-point scale for selection. And what we did in Hilo is we went to a 3-point scale.
Okay.
So you have 1, it's crap. 2 is average. 3, it's above average. I was like, if it's crap, if it scores on anything in a 1, it's out.
Call it.
It's no matter what, we just pull it out. It's done. If it's a 2, that's fine. What we're looking for is everything to be 3, but you never find everything 3 and all that's enough. But as long as there's 2s and 3s, then that's fine. But we're looking for the extreme best. We're not looking for, oh, this one has 0 mites, but it only grew to 5 frames of bees.
Right. Yeah. Exactly.
So if they know honey, who cares? That's
Yeah.
And we got some. Myself saw some of those in this selection. It's no mites, no in Hawaii with drone comb all year long. No mites. But you look at the honey production, it's like no honey. And there's like,
yeah, I think as Tom Seeley's bees, whenever I hear that description, but I think you can get all over the place, but like I could see how natural selection would give one that doesn't raise a ton of brood. If they don't raise a ton of brood and they've got some level of resistance, that it would give them an edge, but they're not commercially viable. It's great they didn't die. I mean, that's a good thing, but I mean, everybody, or at least the commercial guys, are going to want something that actually you can split off of and makes honey too. We want the best of both worlds.
Yeah, I mean, naturally, honeybees can live with Varroa because all they're looking for is, can I live long enough to the next year to swarm?
Exactly.
To produce the next generation. And that's all the honey I need to produce. That's all I have to tolerate the mites is to be able to do that, and I'm good.
Yep, exactly.
I don't have to eliminate all the mites. I don't have to produce 500 pounds of honey.
Right.
We're the ones that push them to those extremes.
Exactly.
Which we can. I mean, we can, and it's fun. I mean, honestly.
I agree.
I mean, it's amazing to see what you can do with just a little bit of selection, but
absolutely.
The more traits you select for, the slower the progress is. And if you select for one trait, you might be selecting against another trait.
Exactly. There's always a trade-off.
Yeah. And so
well, that's what in Harbo's breeding group, he made it known up front. We're selecting for VSH. Like, don't squirrel on ala grooming or anything else. Like, just we're going to keep it focused on this. And I know he did that too, like, because a lot of people get hung up over the color of their bees, and that was me for a long time. But John's like, I don't select for color. I select for VSH.
Yeah. Yeah. And Paul line, we selected for color because when we first initially did that, everybody wanted yellow bees.
Yeah. I'm prone. I love the blonde ladies. Honestly, they're beautiful. The queens are easier to spot if you're selling queens to newer beekeepers or anybody. If you really want to find that queen, the darker girls are a little bit harder to pick out on a brood frame, than the big blonde ones.
Yeah. So the pole line is yet lighter, and of course I
Yeah.
have cordovan in there, and it's yellow. But the Hilo bees we don't select for color for anything.
Okay.
I mean,
What
and
do
I
they-
don't select pole line really for color. I might like select the lighter drones at first,
Yeah.
something like that, or if it's a black queen, I'll toss it out instead of
Hmm.
a striped or a yellow one or something like that.
Yeah.
But Hilo, we don't care. It's whatever. I mean, they have some cordovan mixed in because they got pole line mixed in,
Hmm.
but they can range the gamut of yellow to black.
Yeah.
But most people, if they say they let the bees go, they tend to be more darker than they are lighter.
It seems to be the case. Yeah. I know, like,
Yeah.
if I naturally mate mine around here, I'll start getting some darker ones in there too, which I
Yeah.
had predominantly all Italian looking bees. But, since I was bringing in some Harbo stock a couple years ago, it was kind of I had a few darker ones in there. And
Mhm.
I didn't care as long as I was getting high VSH scores. I kind of let go of the color thing, which is still kind of cool. If I didn't have a day job, I'd probably try to do like Latshaw and have maybe a darker line
No, I know.
and a lighter line too. But I just don't have until I can find somebody to pay me to just play with bees. Haven't found that yet, or at least pay me what I want to be paid.
Well, the lab is going to be hiring for 3 technicians here.
Really?
Time. Yep.
Excellent. Well, I'll have my master's in entomology done here in May.
Yeah, I don't know what the requirements are yet. I know one's definitely going to be like a basic field technician,
Yeah.
straight up. The other ones, I'm not sure, how much lab versus field that they're going to be.
You're gonna want.
But we keep hearing rumors that they are in the works.
Cool.
Because we've had a lot of retirements. I mean, a lot of people have retired over the last 3-4 years, and so it takes the government a little longer to
Oh yeah.
process the paperwork on this stuff, and the pandemic didn't help. That kind of sped up retirement, the slow down paperwork.
Definitely.
So we're hiring 2 new scientists and 3 new technicians this year.
That's awesome.
So it's going to be a full lab again. But yeah, there's 3 technicians. So if anybody is interested, you basically get to play with bees all day.
That's attractive.
Well,
and have retirement too if you're just playing with bees on your own.
Retirement, insurance.
Yeah.
My wife owns her own accounting firm, and so I literally work because I like it, and the insurance.
Yeah, that's awesome.
Basically, I joke around. I was like, when can I quit work? When can I retire and just stay home? And she's like, when we can both get Medicaid.
Yeah, exactly. Yeah, when we don't have to buy
insurance. You're the one providing insurance.
Yeah, that's where I'm at now. My wife, she does okay, but she doesn't have insurance offered there, so I do.
Yeah.
And I mean, I don't have to leave the house. I work remote too, so and I can I take off every Monday, like end of March. Like next Monday is my first of the series. End of all through April, May, and then half of June, and then every Monday I'm grafting, and then I'll tie the insemination schedule in on top of it. But it's a busy life here.
Yeah, sounds like it.
Well, Garrett, I've kept you 3 hours here, man. I
just noticed that.
Yeah.
my earbuds just beat saying, okay, we're starting to die. You need to finish this up.
Yeah, I can't thank you enough. I enjoyed the heck out of this conversation. I hope maybe I can catch up with you at the conference and at WSU. Definitely. I am going, so if you can make it up there and get the travel cleared, maybe we can catch up there. Or if not, maybe we can do another Zoom in a little while and get an update on the Hilo project.
Oh yeah, that'll be good.
Awesome. Well, thank you so much.
Thank you. It was fun. I enjoyed it.
I did too.
My wife's probably gonna be, man, that took forever.
Yeah, well, my wife's like, he's probably going to be up there for 2 hours. Like, no, 3. I probably better go check on her. Well, thanks
Okay.
again.
Yep. Thank you. I had fun.
I did too. We'll catch you later on.
Okay. See ya.
Bye bye.
Bye.